Good morning, everybody. Thank you for joining us today. Today, we are going to be doing the non-addictive pain therapeutics development webinar. We have what I hope will be a very interesting group of presentations and also a lot of time allotted for discussion today. So thank you for joining us. I'm going to give just a really brief intro, and then I'm going to turn it over to Rob Giroux to talk about the kind of broader HEAL Initiative strategic planning that's going on right now and that this webinar is part of. So non-addictive pain medicine development, I think as everybody who has joined here today probably already knows, is a real cornerstone of the HEAL Initiative. In the last several years, there have been 12 funding opportunities created and more than $520 million in funding has been allotted to non-addictive pain therapeutic development. I think there has been a huge amount of success in this area, and one of the major things that we want to try to do today is to give some presentations that have some forward-looking ideas about how we can continue to have success in this area. And I think we all recognize that we really need these new therapeutics to be developed to help people that are suffering from chronic pain disorders, and also to get feedback from everybody who's joined the call today. So again, I just want to reiterate that this particular part of the HEAL Initiative has seen a lot of success. We in the programs that were funded in the first 5 years, we've had at least one, but I think there have been more investigational new drug applications that have been filed and approved. So we've seen drugs that have been developed through this program have moved from the discovery phase into the clinical development phase, which I think is absolutely remarkable. And we've also seen successes in the device area. So it's not just in the drug development area that we've seen things start to progress in the clinical development. So now I'm going to turn it over to, I also want to point out that the co-chairs of this are myself and John Markman at Eli Lilly. John is here. He's going to introduce the kind of structure of what we're going to do today. After Rob Giroux, who I'm going to turn it over to right now, is going to give the kind of overview of the strategic planning for the HEAL Initiative. So Rob, take it away.
All right. Thanks, Ted. How are we looking there, folks?
Looks good, Rob.
Okay, great. Well, thanks everybody for joining us for this very important process. I'm just going to kind of frame the background a little bit and explain the process of where we are and where we're headed, so everybody's on the same page. So the HEAL Initiative, as you know, this is an NIH-wide effort that's looking to speed scientific solutions for the overdose epidemic, and including opioid and stimulant use disorders and the crisis of chronic pain, which is why we're here. It launched in 2018. As you can see there, it's resulted in over 1,800 research projects in every state, totaling over $3 billion total. It's an incredible investment in this effort. It involves collaboration across 19 institutes, centers, and offices at the NIH. Everybody loves a mission statement, so here's the part of HEAL that is related to pain. So HEAL Pain Research aims to reduce pain and the risk of opioid use disorder by developing safe and effective pain treatment and prevention strategies to improve quality of life for all people. So keeping that in mind, the goal of what we're doing here is to come up with new priorities to be forward-looking for what HEAL should be doing going forward. So Ted kind of summed up the progress to date. Many of those initial programs, the first few years, are kind of wrapping up or moving into the next phase. And our goal here is to recommend these new research priorities, as I said, and to really focus on what we should be doing going forward. So this whole process is a strategic planning process. There's a separate process happening related to addiction and substance use disorders. This is specific to pain, and this is the larger effort here. So there's a strategic planning executive committee. I'm co-chairing this along with Kathleen Sluka and ably assisted by our lived experience advisor, Michael Falcon. You can see here the 7 different areas, the subcommittees who are focusing on different aspects of pain that are kind of tackling different aspects that's similar to what we're doing here. This group, Non-Addictive Therapeutics Development. I'm not going to mention everybody that's involved, but you can see the 7 different groups: health, equity, and pain across the lifespan, our committee here today, pain biomarkers and predictors, optimization of interventions to improve pain, implementation in health services, the intersection of pain and substance use disorder, and pain research, workforce, and training. So just like we're doing here, every one of these subcommittees has taken information from previous requests for information and has formed a subcommittee that is holding a similar workshop. Here are all of the workshops and the dates where they sit right now. There's a link here on the screen and the code what you can scan if you'd like to, but basically you could go to this link and see the content of what's happening there, what the dates are, and register for those workshops. And we have a lot of people who are attending across multiple workshops, which I think is going to be very useful. So the goal, okay, so where are we? What's all happened and where are we going from here? So right around now, so we've taken the input that was received. There was a request for information that was sent out. We received about 100 responses from that from across a broad spectrum of stakeholders, researchers, healthcare providers, professional societies, people with lived experience of pain, saying where they think the priorities should be. And that partially helped inform the subcommittee development, but it's also that information has been ingested by these subcommittees and is part of the basis for the discussion that we're having. So these workshops and that RFI should be wrapping up here by early December, hopefully. In January, these subcommittees will be sending back kind of refined, prioritized areas of interest across all the subcommittees to the executive committee. And the executive committee is going to meet in January and kind of help boil these down to draft a set of HEAL pain research priorities that would be presented to NINDS Council for input on that. In March and April of next year, NIH will then develop and publish the HEAL pain strategic plan. What that is doing, the reason that the timeline here is so short is the goal is to get this all done in time to develop concepts and funding opportunities for the FY27 NIH budget, which we're hoping will have lots of opportunity for pain research going here. So a couple of things. This webinar is being recorded. And the chat, there is a chat and a Q&A that we ask our attendees to use freely. This is the way for everybody to be engaged in this process. Those of us on the committee here will be monitoring that and raising some of those questions during discussion throughout the day. The recordings will be available for a period of time after, so folks can rewatch, or people who can't attend can watch it. And then there is still opportunity for further input if you've kind of processed these discussions a little bit and want to give further input. You can see at the bottom here this HEAL question at od.nih.gov. And if you include HEAL pain strategic planning in the subject line, that'll help us kind of make sure that information flows to the right location. So that's the process. It's a rapid one, but this is important stuff, and the NIH takes this input very seriously. And this is really an important way for the community and for us to hear from all stakeholders to make sure that we're not missing anything. What have we done well? What have we missed with the first phase of HEAL, and where should we be going in that next round? So with that, I will turn it back to you, Ted.
Thank you very much, Rob. And I'm now going to turn it over to John Markman from Eli Lilly, who's going to talk about the overall workshop logistics, and then he's going to introduce our first panel, which will be the lived experience perspective. So John, over to you.
Thank you, Ted. Thank you very much. Good morning, everyone. My name is John Markman. I'm a here at Eli Lilly involved in analgesic and drug development. I've also had the privilege as a neurologist and clinician and investigator to be part of the EPICNet clinical development program as part of the first set of the HEAL initiative efforts. And so I've really been a privilege to be part of an enormous scientific effort focused on innovation to bring safer, more effective pain relief to patients. And today's meeting, like the HEAL initiative since its inception, is really, I think, a reflection of the NIH's willingness to listen to patients, clinicians, and scientists in the name of innovation and doing it in a transparent way. So it's really a privilege to be here today with everyone and introduce this panel. So the focus of today's 4-hour session will be sort of broken into 4 areas. The first will focus on the importance of basic science discovery. But before we launch into that, we're going to hear from 2 patients who've lived with chronic pain. And I think that will really ground our discussion and give us our focus about where we want to go with the rest of the agenda and really give us a North Star. So we'll focus on the importance of basic science discovery. We'll look at disease-specific mechanisms of discovery and disease modification and have a panel on that as our second set of activities. Then we'll turn to therapeutic discovery for pain. And then lastly, we'll look at small business innovation. And the way this will be set up is a series of presentations followed by a panel discussion. And you all have the opportunity to add your comments and into the chat. And we will feed those into discussion as we contemplate the talks and the presentations throughout the day. There'll be a break in the middle of the day from 11:40 to 11:50, and then we'll be back with you for the last 2 hours of the session. So that's the contour of the day. And without further ado, I'll turn it over to Will Schwabel and Christy Fishman, 2 patients who can tell us really about what it is like to live with chronic pain and what we need to focus on as we think about planning a course for discovering new therapeutics, both from the basic science perspective as well as in clinical development. So just it's an incredible privilege to be on this panel with such an esteemed group of scientists, but also to be here with Christy and Will. So again, look forward to their talks. Thank you so much.
Hi, everyone. I'll start. My name's Christy Fishman. I'm a medical malpractice attorney by trade, although my true passion is as a mother to my 2 kids. I'm here today participating on this subcommittee as a person with lived experience. My pain is neuropathic and musculoskeletal in nature. As a bit of background, my left spinal accessory nerve was inadvertently severed in 2013. And despite several surgical interventions, I never regained proper function of the trapezius muscle on my left. And so as a result, I have musculoskeletal pain in my neck and upper extremities, along with chronic headaches and migraines. Additionally, the surgical attempts to address the severed nerve and reintegrate that muscle caused consequential damage and dysfunction to other peripheral nerves in the area, which left me with complex regional neuropathic pain in my neck, left arm, and left hand. And so that'll give you a little bit of background with respect to Will and I, what we talk about with respect to what we would like to see or make sure is considered in the studies and in the funding that's put forth for the scientists doing the studies to, further along and help develop different modalities and medications to try to treat those types of pain. So I'll turn it over to Will now to introduce himself.
Thank you, Christy, and I'm very honored to be part of this. My name's Will Schwalbe. I'm an author and editor. As an author, I wrote a book called The End of Your Life Book Club about the books I read with my mother when she was dying of pancreatic cancer. So I've also been a caregiver to someone experiencing chronic pain over the course of several years. I was hit with a blizzard of strange symptoms in 2016. My feet and arms were sometimes freezing cold, sometimes burning hot as though I had a terrible sunburn. I had shooting pains that were sort of as if a dentist were doing something without Novocaine on your teeth. Pain at touch, even the sheets on my feet were so painful that I had to and still have to sleep with my feet outside the sheets, as well as a lot of autonomic symptoms, inability to digest food, sweating disorders. And I was very lucky after 6 months of just not knowing what was going on to find a neurologist who diagnosed it as small fiber neuropathy, which is a pure small fiber neuropathy. It affects probably around 175,000 people in the United States at the moment. It is able to be diagnosed with a skin biopsy that gives over 90% certainty that that's the thing. So I was positive for that. I did want to mention that, you know, as someone living with chronic pain, it's not getting better, but in many ways I'm getting better at living with it. But that involves a full and happy life, as well as many days when I'm confined to bed. Thanks to the Family Medical Leave Act, I'm able to stay home without pay on the days that I need to, as well as an understanding workplace. And I am a long-term taker of medicines. I take gabapentin, which does a lot for me, but increasingly less, and a pill called Mestinon pyridostigmine bromide. I mispronounced that, I think. And that also has proved very helpful, but it's a ongoing condition, and I'm passionate about the work of this committee. I do have a prescription for opioid pain medications for breakthrough pain, and I try very hard to use them as infrequently as possible. But I definitely sympathize with other people with painful conditions who really feel they need those medications, and that's the choice they're given. So, Christy and I, having introduced ourselves, we're going to move on to a couple of more general topics, and have a couple statements, and then go right into them. Christy?
So one of the things that seemed to come up during a lot of our conversations, both as lived experts kind of crossing the different committee lines and during our individual committee meetings, was the impact of side effects from the various medications, whether they be opioid or non-opioid pain medications. You know, a lot of them are focusing on the nervous system, and naturally there's a lot of side effects to the nervous system that you know, can be very detrimental or helpful depending on the patient. And you know, so through our participation, we thought it would be helpful to provide, you know, an overview of some of those considerations. Sometimes they can be so debilitating that it often makes a patient question whether, you know, they'd rather just deal with the pain than to take the medication to have less pain, but then the quality of life or their ability to be productive in society takes such a hit that, you know, it kind of raises the question whether taking those medications really works. And so the side effects of drugs should be something that's considered, and the outcomes of studies should incorporate those kinds of considerations as opposed to strictly focusing on the impact on the pain levels.
And one of the things that Christy and I discussed about the side effects is they often force people with chronic pain who are taking medicine with side effects to take an ever-increasing array of medicines to treat the side effects, both prescribed, over-the-counter, and maybe even illegal, so that it makes it a really hard job for us and our doctors to figure things out. The first one we wanted to talk about was sleep. And the fact that for many people I know and me, sleep is the big story. That because I have a painful condition that keeps me up at night, it leads to lack of sleep. It leads to feeling horrible during the day, and my health decreases. Or I add Ambiens and gummies and various things to try to get me to sleep, which cause their own problems. Christy?
Yes. And so on the flip side of things, as much, you know, no doubt everyone's quality of life improves when they get adequate sleep. And anyone with chronic pain will tell you that that is a challenge in and of itself. But at times, taking the medications that help us obtain that sleep makes someone sleep in such a depth that it can interfere with what their responsibilities. For instance, you know, for me, I often chose just to go with interrupted and you know, short stints of sleep, basically probably from exhaustion, but because I was a single mom at home and I had 2 small children, so I couldn't take medications to help me sleep and then risk not waking up if they needed me, or, you know, to take care of my own responsibilities at home. And that kind of goes to quality of life and, you know, productivity. And so while it's a blessing to some, it could be a curse to others. And so it's basically what we're saying is it's something that needs to be thought about, not just kind of an afterthought as, oh, that it makes people drowsy, so we should warn them about that. You know, it trying to incorporate that and find things, you know, that don't have that side effect, or, you know, that have that side effect intentionally for someone like, you know, Will, that can afford to do that. So
Yeah.
go ahead.
I mean, we use the example of, we were sort of joking when we were talking about this, that if you have a cold, there's Dayquil and NyQuil. And even for a cold, they recognize that people really need to distinguish between medicines that put you to sleep and medicines that may keep you awake and ruin your sleep. We also wanted to talk about other side effects. And again, I'll shoot it back over to Steve.
So you know, I alluded to it earlier, but the cognitive effect of a lot of the pain medications, you know, as an attorney taking the medications, it was also, again, it was a choice that I had to make, whether I took the medication and was sharper, you know, or had less pain, so I, you know, could pay better attention and focus. But at the same time, it would have an interfering effect because, you know, it would make me have trouble you know, calling words and recall and short-term memory issues. And so, you know, it was kind of choosing between two evils, so to speak. And so the cognitive impact on, you know, these, especially neuropathic pain medications, right? And Will and I talked about maybe trying to have more of a focus on distinguishing between peripheral nerve pain and, you know, a pain that addresses the core nervous system, you know, with the brain and spinal cord. Because again, for me, that was my experience was, you know, if I was on Lyrica and very, very high doses of gabapentin, and, you know, I would go to court and struggle significantly to remember what I had just said to the jury, nonetheless, you know, not repeat myself and not draw a blank of that one word that I say 100 times in a day, but I can't think of it at this moment. And so it would interfere again with quality of life, you know, ability to do the things that you need to do in a day.
Yeah, and I had very much the same situation. At one point, I think they put me on Topamax, I think it was called, which patients refer to as Dopamax. And I'm a writer, and it's simply unacceptable to be on a medicine that causes me to lose language. I wanted to talk also about the side effect of weight gain. I was on Lyrica too, and put on 15 pounds, you know, what felt like overnight. And that's a very serious side effect for a lot of people, quality of life, overall health. And when you think about the number of people willing to take extreme side effects to lose weight through something like Ozempic and those populations, I think weight gain is often trivialized as a side effect, and it's something that is a major effect on people's quality of life.
Yeah, I would agree with that. And, you know, it affects all of these things really trickle down into mental health as well. You know, I mean, living with chronic pain is challenging enough as it is. And I say to many people, you know, I take it one day at a time. You know, you wake up and you just deal with the challenges, adapt and overcome. But adding challenges to that, such as weight gain or the cognitive effects or, you know, drowsiness, things like that, it just makes it harder because, you know, it's exhausting. It's an exhausting thing to live with. And so, you know, having to tackle all these other issues, it can be very difficult.
Yeah. And we just wanted to address one other category, and I know we're getting close to time, so we're going to do this in fairly short order, but which is the difference between medicines that have been specifically developed for chronic pain versus short-term pain. And to that, first, Christy, I think you were going to talk about the pain level thing.
Yeah, no, and during one of our discussions, we talked about, you know, when you walk into a doctor's appointment, especially a neurologist or a pain management appointment, as a person with chronic pain, the first question they ask is, you know, what's your pain level today? And although it seems like a simple question coming out of the nurse's mouth, it's a very complicated question for many of us. And like, for instance, for me, you know, there's so many different types of pain that I have with the musculoskeletal. Are you talking about my headaches? Are you talking about the neuropathic pain in my hand? Because granted, all of the levels and you know, whether that's improved or it's deteriorated is relevant to management of the pain. And so, you know, using such a simple, you know, kind of archaic scale for chronic pain, I think is problematic and, you know, should be considered in terms of going forward and trying to be successful. It's similar to the outcomes of the studies. If you're simply asking, you know, your patients, you know, was your pain level improved? You might want to provide more ability to expand on that.
And I wanted to talk a little bit about the problem of titration and maybe paying attention if possible. I know this is a massive wish list task of science, but on medicines that don't have extreme withdrawal symptoms. So for example, as someone on high dose gabapentin, at certain points, I've wanted to try other medicines. It's taken me a month to 6 weeks to taper down safely from gabapentin. I mean, maybe less depending. Then if I take a drug that needs to titrate up, it could be a month to get to dose on that. And then I have to spend a couple of weeks on that to see if that actually has a better effect. And if it doesn't, then I have to titrate down from that. And then I have to titrate up from gabapentin. So it can be months of really diminished quality of life just to see if another drug works better for me. And I know a lot of people who experience chronic pain, it's so daunting, they just stick with whatever they've got or go to opioid or other pain medications to supplement.
I think lastly, the one thing that we wanted to mention was, it came up in a few of our discussions, was insurance, you know, and this may not be necessarily where things can be directly impacted, but, you know, the insurance companies look at these studies and, you know, determine whether things are effective to treat and off-label and on-label. And so, you know, the more studies to help with developing modalities to treat chronic pain, obviously the more likely that some of us will be able to actually use those medications. You know, the ones that they may be effective and they may be great and have no side effects, but if I can't afford it, then I'm not going to be able to take it in the real world. So.
Yep. And then just to end with a couple of statements, which is we're really excited about, for example, things like the next panel. We need more basic science. We need to understand the mechanisms of pain better. That's very clear.
Yes. And side effects, you know, the things that we talked about, again, they shouldn't be a side issue for chronic pain. They should be a central issue. And, you know, whether they're positive or negative, they should be considered in the studies and not just an afterthought. So I think that about wraps it up.
That's it. Yep.
I'm not sure. Are we turning it back over to
so
you,
much.
Ted?
I think that was at once
Actually,
poignant and lucid in so many ways. So I just I think that sets the stage in such a compelling way. So thank you both for sharing that. It is really helpful. Ted, I think you're going to turn it over now.
I am, however, we have a little bit more time, and there's actually a question in the Q&A. So, Christy and Will, if y'all are willing to take a question or two, maybe we'll do that before we turn it over to David Julius. So the question is from Simone Hartunian at WashU. The question is, and he says, first of all, enormous thanks for sharing your experience. Considering that no ideal drug may be developed, if you could choose one side effect, drowsiness, sleepiness versus cognitive impairment versus weight gain, for a benefit of 50% or so pain relief, which one, if any, would you pick?
Okay. If you want me to go first, Will, again, to Will's point, weight gain's a tough one to fight. So I definitely would not pick that one. You know, I would say probably drowsiness in hopes that, you know, could time the medication to, you know, have that be less of an interfering side effect. You know, obviously for me, my kids are older now than they were when this all started. And so, you know, drowsiness and having a little bit more deep sleep, they can get out of their beds and actually come get me instead of me needing to hear them. But, you know, the cognitive impact, it's just for if I can't control the pain during the day, it's going to interfere with my ability to be an attorney. And so if I take the medication and it addresses the pain 50%, but it also takes away 50% of my cognitive functioning, like it's a trade-off that, you know, it's just, for me, it would probably be drowsiness that I'd be able to tolerate at this stage.
I totally agree with Christy. The weight gain, I think, is often trivialized, but its overall impact on health and well-being is huge. And the cognitive impairment, if I'm not who I am, then that's a crisis.
Let's see if we have any more questions. Don't see any more. Oh, yeah, actually there is another one. So we got them coming in a little bit in the chat and also a little bit in the Q&A. What is your experience with topical agents to treat pain?
I'll jump in on this.
Yes, Will, I think you should.
This relates to what Christy said. It's very frustrating for me because lidocaine 6% patches, when I am in a pain crisis in my extremities, and I can slap them on my feet, have an enormously beneficial effect. My insurance company will not cover them. They say they're off-label for small fiber neuropathy and not approved. One of the top neurologists at one of the top hospitals in New York attempted to get the insurance company to reverse that decision, and they won't. So this quite effective thing, which cost me $600 a month, I have to ration because it's just too expensive to use whenever I need it. And I would love more investigation of topical for peripheral, especially small fiber neuropathic pain. I did want to mention one thing in the comments, if it's okay if I chime in. I love the way that you've asked separately of Christy and me about the side effect thing, because it is very personal and people do have very different things. And the more choices, nothing will have no side effects, but the more choices we have, I think the better off we'll be.
Yes, agreed.
Christy, did you have any comment on the topicals?
Oh, the topical. The topic for my neuropathic pain, the topical medications did help a little bit. Actually, Dr. Markman, we used it was capsaicin, I believe we tried. We tried some capsaicin treatments to try to numb the nerves. And the pain that those treatments came along with was sometimes difficult to tolerate, especially in the days following when hot water would hit the area, whether I was washing my hands or taking a shower. But there was some benefit. I've never had the patches. I've never used the patches. I did have some lidocaine cream. I didn't really notice much of an improvement with that. They have not had much improvement with respect to the musculoskeletal pain. You know, the knots and the imbalance and the pressure that it's putting on my neck and the occipital part of my head and stuff. We did use a little bit of I can't think of the word. Dr. Markman knows all of this probably better than I do, but topicals were not that helpful to the pain that I experience overall.
Do you mind if I answer one of the questions that came up on the side? I don't want to run over our time.
Sure. We're not over time. We got time.
Someone asked about the timeline between first symptoms, diagnosis, and intervention. I was incredibly lucky. As I said, mine was 6 months. I went first to see a cardiologist. Then I went to see a vascular specialist. They had no idea what it was. I was very lucky to find a neurologist who was able, 5 minutes into my spiel about my weird, crazy symptoms, was able to diagnose it and then order up the skin biopsy. But I will say that it's been my experience that people with small fiber neuropathy, but a whole host of other conditions, have sometimes gone years or even decades without a diagnosis. And I've noticed, not an original observation, that especially among women patients, that doctors often of every gender are very quick to dismiss this kind of neuropathic pain, odd bunch of symptoms as, psychosocial disorder.
And I'll just comment on that as well. I think there's a little bit of a disconnect between doctors and, people trying to diagnose this when it comes to objective versus subject complaints of pain. For me, the nerve was severed during a medical procedure. My symptoms developed instantaneously when I awoke from the surgery. And so, diagnosing the severed nerve and then diagnosing the cause of the pains that kind of surfaced each time there was a different intervention attempted to try to solve that problem. I was fortunate enough, I guess, in that way that it was quickly, attributed to, well, we just did a 9-hour nerve transfer, in that region, and we took a branch of the median nerve, and now she's having severe neuropathic pain in her along the median nerve outlet. So, that makes sense. There was sensory and autonomic nerves, and obviously in the transferred nerve, and we cut those and, transferred them and damaged them. And so it makes sense. But I think if I was just a patient that walked into a doctor's office and complained of the pain that I have, and didn't have that explanation, that very, clear objective proof of what had happened, I think I probably would still be trying to find help.
So we have
And
one
then there
more
was another
question.
comment just
Go
below
ahead, Chris.
from Dr. Markman, and I think
Yeah.
you and I talked about this during many of our discussions of medications and the side effects. And absolutely, I wholeheartedly agree and think that working on peripheral nervous system with few side effects would be, a step in the right direction.
Yep.
Well, speaking of the peripheral nervous system, maybe we can move on now to the next session, where we will have a number of people on the panel who have a strong expertise in the peripheral nervous system, including Dr. David Julius, who needs really no introduction, but will be giving a talk here. So that the topic of this next session is why basic research remains an essential first step in assessing and treating chronic pain. And we will hear a short talk from Dr. Julius. And then our panel will be Dr. De Silva, Dr. Malfay, and Will Schwable will again be on. And we'll hear some thoughts from Dr. De Silva and Dr. Malfay, and then Will, you'll be around for commentary. So with that, I think I will turn it over to Dr. Julius to give our presentation. David?
Okay, thank you, Ted. And thanks everyone for including me in this important discussion. I wanted to start, I guess we were asked to say something quickly about, how or why we got involved in pain research. And I'm a biochemist and molecular biologist by training, but what really has gotten me interested in pain initially was just my general fascination with the basic biology of sensory systems, because it's really our sensory systems that enable us to experience and appreciate the world. And at the time, there was a lot less known about the pain pathway, nociception and pain, compared to other sensory systems. So that just drew me in as a curious scientist. But aside from that, I think we all recognize that there's so much need and therapeutic potential in studying pain that was sort of the, cherry on top in essence. And we all know that that's, I often say to people that of our senses, I think the pain system is the one that's most necessary and critical for our survival and well-being. And of course, as we've heard from Christy and Will, it become maladaptive, as we say. Generate a whole host of other problems. So there's fascinating tremendous therapeutic need. All right, so about the importance of basic research. And I would say that, for all of us, where there's an urgency to cure a disease or come up with treatments, sometimes we want to skip over the basic step of basic science, and that often goes on the chopping block. But I thought I would start by just showing 3 examples of where beautiful basic research over many years was essential to developing drugs and therapies that have really been tremendously impactful for human health. So let me share my screen. Okay, let's see. Can everybody see that?
Yep.
Okay. So I just thought I'd lay out these three examples because I think it really sets the tone for how important basic science is and the impact that it can have, and really how it's essential to taking what I call sort of a mechanism-based approach to medical breakthroughs, which I think is really the only durable way that modern science can really lead to new cures. So one that we're all pretty familiar with is the use of statins to control hypercholesterolemia and to protect from heart disease, cardiovascular risk. And the ability to, the statins really are an outgrowth of many, many years of basic research, understanding, as I just showed in this simple diagram, the biochemical steps that lead to cholesterol biosynthesis and the uptake of cholesterol from the environment. And there's, decades of cell biology and biochemistry that went into this pathway to understand that statins inhibit this enzyme called HMG-CoA reductase, which is a rate-controlling enzyme for cholesterol biosynthesis. And this work by Brown and Goldstein and others really laid the groundwork for understanding that levels of LDL cholesterol are a major risk factor for cardiovascular health, and led to this very targeted approach to use statins to control hypercholesterolemia. Another, I'd say, a more recent but really incredible example of how important basic research is to developing rational and targeted therapeutics is a group of drugs that have recently been developed, which are almost essentially a cure or a very effective treatment now for cystic fibrosis, which is one of the most common inherited disorders. And as many of you know, it was basically until recently fairly lethal disorder for people at a relatively early age. And years of attempts to identify the gene through human genetics led to the identification of this protein called the CFTR, or the cystic fibrosis transmembrane regulator, which was the causative gene in which there are mutations that lead to cystic fibrosis. And years of research on this asked basic questions. What does the CFTR protein do? How is it defective in people who have genetic mutations? It was determined that the CFTR protein is a chloride channel, which explained many of the phenotypes associated with cystic fibrosis, namely the inability to regulate solute transport across epithelial cells. And more importantly, it was determined that there are basically two types of mutations in most people who have cystic fibrosis, one minor class called a gating mutation and another called trafficking mutations. And this really elucidated what the problems are in the protein and why people who have cystic fibrosis have problems at the molecular level. And through an amazing series of experiments, again taking years, people at drug companies developed this, mostly Vertex, this collection of 3 drugs that actually correct these defects sequentially. So 2 of them bind to the protein and correct what's called a trafficking mutation that now allows this protein to get to the cell surface in people who have the most prevalent form of cystic fibrosis. And the other drug corrects the gating mutations, which basically means it makes opening of the chloride channel much more efficacious in people. And these 3 drugs together have basically now almost cured cystic fibrosis for a vast majority of people who have these most common mutations. The last example that I wanted to give is in the area of cancer biology. And this is, again, another beautiful example that has saved many thousands of people's lives. It's a drug called Gleevec, which inhibits a tyrosine kinase called the c-Abl kinase. And through many years of basic research, again, it was determined that people who have this form of cancer called CML have a chromosomal translocation that leads to the aberrant activation of this receptor tyrosine kinase. And by studying this protein for many years and understanding what its structure looks like at 3 dimensions, chemists were able to design very specific drugs that bind to the c-Abl kinase and only to the c-Abl kinase among the many kinases in our body, and inhibit this protein in people who have an aberrant form activated in CML. And this drug is about 90% effective as a cure for this form of otherwise deadly cancer. And I just have this one quote that I put up that I thought was quite beautiful. If I can get it, there we go. From Nature Education, that said, the development of Gleevec taught scientists that by understanding the biology of a disease, it is possible to learn how to treat or cure that disease. And without this basic research, none of these drugs would be available to us. And the last thing that I want to say about this particular slide is that although basic research elucidated the mechanisms leading to disease, it took many years to find the drugs that worked, even when we knew the targets. So the watchword here is really that we need to be targeted, but we also need to be patient. That the first few drugs that were generated against many of these targets did not work or had side effects, and it really required the persistence of investigators and chemists and pharmaceutical firms and basic researchers to use the basic science that has come out and to really make drugs that work well for these different types of diseases. But I think that they're both very inspirational in terms of showing us how dedicated research, sometimes just curiosity-based research, can lead to cures that have really had a major impact on human health. All right, now with respect to pain, I think we still have, we've known a lot about the pain pathway at many levels. People have been studying pain for many years. It's really only in the last 20 to 25 years that we've taken real molecular insights into this process and really tried to identify molecules and cell types that are involved. And there's really, so much to be learned at every level. Of course, understanding how the periphery detects noxious stimuli like intense pressure, intense temperature, chemical irritants, etc., happens at the periphery. And this process of understanding how the sensory nociceptor works is really a work in progress. We've understood, so much has been learned about this in the last few years, but we still need to know more about the molecules that are involved, how they transduce signals, how they convert these signals into electrochemical signals in the nervous system. And to bring up the point that Christy and Will have mentioned about side effects, many of us think that this is the best possibility to develop drugs that are selective and efficacious without having side effects, because if we can develop drugs that target molecules that are only at the periphery, then we have a better chance for introducing therapeutics that do not have side effects because they don't affect other parts of the nervous system. So that's why personally I'm very heavily invested in understanding how things happen at the periphery where we first detect pain signals, and seeing if we can find the mysteries of those cells and understand what kinds of proteins they express that might be targets for new drugs. Similarly, we know in broad scale how these neurons communicate with cells in the central nervous system, in the spinal cord, for example. We know a lot about the anatomy, but there's still so much to be learned about the molecular identity of these cells, how they communicate with one another, how they send their signals from the periphery to the central nervous system. This is going to be incredibly important for devising more sophisticated, not just drugs, but also devices like neurostimulators that can modulate these pathways. We need to know a lot more And finally, of course, pain is a cognitive process, emotional, etc. And we need to learn more about these pathways in the brain. This will be incredibly helpful for obtaining subjective assessments of pain, objective assessments of pain, using imaging, so on and so forth, and in all of these pathways for developing what people would really like in terms of objective assessment, namely biomarkers, which we are very far from because we really haven't identified yet through basic research reasonable biomarkers for pain. All right, the last point I want to make is that we often use this one term pain like we do cancer, but if we've heard from Christy and Will, pain is a multimodal mechanism. It affects different tissues and organs in different ways, and it likely involves different mechanisms. We need to understand the mechanisms for different types of pain if we are going to be intelligent about taking mechanistic approach to deal with these. Migraine pain certainly has different underpinnings than, say, visceral pain or osteoarthritic pain, which we'll hear about later. And basic research will tell us, or hopefully will give us clues as to how these different pain syndromes are the same, but importantly, how they differ. And I'll just show two examples here of, obtaining mechanistic insights, in this case, into different forms of visceral pain. For example, work from a group at UCSF that involves me and folks in Australia have identified a lot of new cell biology in the gut that is underpinning the role of different cells like enterochromaffin cells in driving visceral pain, and how this differs in men versus women, which is a critical issue. And more recently, I chose this publication from the literature where there's an interesting elucidation of a possible role of this peptide called CGRP in driving pain in endometriosis, which is a very poorly understood type of pain, but affects millions and millions of women. And this basic aspect of research has already suggested that drugs that are already in the market to treat migraine, for example, that interfere with this pathway of CGRP signaling, may actually be efficacious in treating endometriosis pain, which is something that we had no idea about just a few months ago. So in summary, I'd say we've learned a ton about how the periphery works. There are so many molecules that we and others have identified, but there's still so much to learn. And I think we have to be patient that while some of the drugs developed against these receptors and ion channels have not shown promise yet, I think we have to think about the examples that I talked about earlier, where even knowing the mechanism and the molecules involved, we have to search for the right drugs that affect these, and that may just take time, but I think it's well worth the effort. This will all depend on the power of basic research. Thank you.
Thank you so much, David. That was terrific. And now I'm going to turn it over to Dr. Joyce Da Silva to give a short talk on her perspective.
Can you hear me? Can you see me?
Yep.
Yeah.
Yep, we got you.
So hi, everyone. I'm honored to be part of this panel and to have the opportunity to hear Will and Christy sharing their experience. That was amazing. And my name is Joyce Da Silva. I'm an assistant professor at the University of Maryland, Baltimore. And my research focuses on the effects of sex and age on brain mechanisms of chronic pain in both humans and rodent models, particularly using imaging, functional MRI, EEG, and also molecular techniques in rodents. And today, I would like to bring attention to another factor that is sometimes overlooked in pain research and funding opportunities, which is aging. We know that chronic pain is far more common in older adults, but we still lack a clear understanding of how aging affects both acute and chronic pain. And this ranges from changes in pain sensitivity, how much pain they feel, how much pain they can tolerate, to the underlying biological mechanisms as well. And we know that this problem won't go away. So life expectancy is significantly increasing, and the elderly population is experiencing chronic pain at higher rates. So for the sake of time, I will stop here, but we have some points for discussion and questions about aging research as well. Would love to hear your thoughts on this factor and what are the barriers to including aged animal in studies, for example, or the elderly population in our clinical studies. Would love to hear your thoughts.
Thank you very much, Joyce. So now I'm going to turn it over to Anne-Marie Malfay.
Thank you.
Thank you.
Good morning, everyone. Yeah, so, and thank you for organizing this panel. And Dr. Julius, I thank you very much for highlighting the importance of basic research. I come from the world of osteoarthritis, and so in a minute, I will talk to you all about that. And I think that is really a field where you will see it's such a tremendous unmet need. It's essentially the major source of pain, one of the major sources of pain in the world, and it cannot be addressed at all. And personally, I think it is because of the lack of basic research that has gone into this field totally. There just has been this, for decades, there's been a complete focus on how can we stabilize the joint, how can we protect the joint from progressing to a damaged joint. But there's almost been no research in how can we actually make sure that it doesn't hurt along the way. And one of the major obstacles has actually been touched on here. The risk factors for osteoarthritis are aging, joint injury, and obesity. So all three of these risk factors are on the rise in our society. And so they are often difficult to model. The easiest one is joint injury. And so all the models up until now have used joint injury to model osteoarthritis. Now that's actually 12% of cases of knee osteoarthritis. And so there's just these, and these are shorter lived models. But when we then model this disease in animals, we don't take aged animals. And so if you take a mouse, it will also be spontaneous arthritis. That hurts. And we've only recently really been able to show that, because we recently only had the opportunity to look so long. So in the pharmaceutical industry also, there is this, where I worked for years, there's this you know, there's the need to progress, and people then will use animal models that are faster progressing. But then that actually, you know, defeats the purpose of looking for the real risk factors, which are slow progression and aging, for example. So that's just one point that I would like to add, that we often hear, well, animal models are not translatable into people. But I actually think it's often because we don't really use sophisticated models that really try to go after the very difficult part of the disease to model. For example, very slow progression or age. So I will leave it at that. Mm-hmm.
Thank you very much, Anne-Marie. So now, Will, I know we don't expect you to give a long talk here since you just did that, but if you have any thoughts you'd like to contribute before we start going to the discussion, we'd love to hear them.
Actually, no, I don't, but thank you for the opportunity.
Sure. All right, so we have a question from Dr. De Silva. So I'm going to take the opportunity to actually turn it over to you, since both you and Dr. Malfay just kind of addressed this. Maybe you can start by giving us your thoughts. I mean, I think it's an extremely important topic. So what Dr. De Silva put in the chat was, what are the barriers? What are the barriers to including aged animals or older adults in studies, and how can funding agencies address this? So Joyce, if you'd like to maybe start us off with what your thoughts are, and then everybody else could chime in on that one, because I think it's an extremely important and obviously underserved topic.
Yeah. So I think I would start by probably mentioning the biggest concern of preclinical researchers. So cost is a major concern that we have when we designed our experiments. And of course, that is related to funding opportunities. And we all know that if we would like to develop an aging model, this animal would be more expensive. If you would like to buy an old animal, it's much more expensive than the young animals. And if you decide to do longitudinal studies with them and follow the disease progression, as Dr. Malfait was explaining, the cost is going to be very high too. So I think this is one of the main issues why researchers are not very interested in pursuing this line of research and looking at age effects. But also there are some opportunities from, for example, the National Institute of Aging, and they could donate the animals to you if you get a grant from them. So I think we need more funding opportunities, specifically looking at the effects of age and long-term progression of diseases, specifically focusing on pain. And we have learned a lot from sex differences, and that was a very difficult transition in the field as well, having both males and females investigated, right? But now I think it's time that we grow our pain field in the aging aspect as well. And just for those who are not from this specific field, it's generally accepted that aging makes both humans and rodents less sensitive to pain, but at the same time, less tolerant and with impaired endogenous pain modulation, or this endogenous ability to inhibit pain. And we know that older adults are more prone to develop chronic pain. They can experience similar or worse pain levels depending on the pain condition. So it's also important that we analyze different pain conditions. And when we talk about treatments, which is the focus of this group, for example, opioids, they are often less effective in the elderly population. So it's a very complex topic, but definitely requires further investigation and because the studies often will show mixed results, right? Because there are just a few studies out there saying in more informal terms.
And Anne-Marie, would you like to add to that at all? No, David?
Yeah, I'd like to say that Joyce has raised a great point. You know, many people didn't study sex differences, of course, because it can double the cost, but certainly aged animals is more of a challenge. And I think part of the problem is that if you look at some of these disorders, like some of the things we've been looking at, visceral pain, you know, the interest is spread across different institutes, like maybe NINDS or NIDDK, and for osteoarthritis, I assume that, you know, also involves National Institute of Aging, and you know, be more effective in some ways if some of these questions were not so siloed among different institutes, but there was sort of some kind of pan-funding mechanism, so that, for example, if you needed to use aged animals, you didn't have to have a grant per se from National Institute of Aging, where there's maybe more of a focus on things like Alzheimer's. And so, you know, something that would sort of bridge, because what we're working on, I think, is pain community, really biology that bridges so many different areas. You know, if you're Anne-Marie and you're working on osteoarthritis, it's going to be NIAMS. If you're, you know, someone like me also looking at visceral pain, it's going to be NIDDK. And so, you know, we're studying diseases and disorders that affect a lot of different physiologic systems. And hopefully there could be a way to make that bridge a little smoother so that we're not sort of siloed into different institutes.
Mm-hmm. And Rob just raised a very nice point in the chat that things being spread across different institutes is a challenge, but this is also an area where HEAL can help by placing more emphasis on areas like aging. And I guess since I'm talking, I'll also take the initiative, just mention one thing from what I think is kind of emerging from the Precision Pain Network, which is also a HEAL-funded set of projects focusing on characterization of the human peripheral nervous system and the CNS to some extent. I think we have seen, and other groups have seen that are involved in that, some changes in DRG transcriptomes of human sensory neurons during the aging process. So I think that probably will be an important thing that will emerge from that project as well, that'll give us clues about how to think about treating pain disorders in older individuals, and why, I mean, in addition to all the other things we know, why older individuals might be more susceptible to developing chronic pain. So there's a question for you, David, related to your presentation that I'll ask, and then I'll go back over to some of the ones in the webinar chat. So this one is, can you provide more information about the new alternatives for endometriosis or IBS patients? And could this alternatives represent a new opportunity for patients with less side effects?
Well, the endometriosis thing's not an area of my expertise, but I just happened to pick this paper out of one that's just recently published in the last couple of weeks. More as an example, I don't think it's you know, on the verge of therapeutics, but it does identify this peptide that we all know about called CGRP that's so important in the peripheral nervous system and the pain pathway, especially for inflammatory pain. As being a player in modulating sort of the immune environment around sensory neurons that innervate the endometrium. And this paper suggests that interfering with that signaling pathway could be beneficial not only for the pain associated with endometriosis, but for the progression of the disease. And the interesting thing there is that drugs that interfere with CGRP signaling are already being used for another pain disorder, namely migraine pain. And so they raise the possibility that, you know, this could be a way of using drugs that have already gone through clinical trials for this other sort of pain syndrome. But I assume that that's going to require clinical trials in and of itself. In terms of other forms of visceral pain, like IBS, something that I've been working on with a great group of Jim Baer and Holly Ingram at UCSF and Stu Brierley at University of Adelaide. This is really an area that affects so many people. It's also important and fascinating because it affects primarily women and likely has a lot to do with hormonal cycles. There's not a lot of effective treatments for IBS. There are some that target serotonergic systems. We and others are now trying to identify more about the cell types that are involved and the receptors and neurotransmitters that they use to signal nociceptive signals from the periphery. And so I think that this is, we've just been working on this for the last couple of years or so. I think this is a real growth area in pain. A lot of people are focusing on pain from within these days in terms of what we call nociception, which is so important, not just cutaneous pain. And so I think that this will bring to light new strategies and targets for treating these types of pains, but it's still sort of early on in the process. But I think the important thing to say here too is that, as I said before, and things that like Joyce have commented on, we need, and Anne-Marie, the important thing here is to develop new models and techniques for studying pain in things like mice, other animals, and really studying them in the context of these different physiological processes. And so I think that's where there's been a lot of progress in the field in the last even just 5 years in that. And so I would keep an eye on that space, and I'm optimistic about where the progress will go from a molecular and a pharmacological standpoint.
So Joyce, you kind of answered one of the questions online, but in the one minute that we have left, maybe you could go ahead and chime in on that. If you'd like to.
So that was a great point, and it's another major challenge is we have to be productive and publish and help our mentees to improve and increase their productivity with scientific papers. And we know that long-term projects could take a long time. So what I would say is that as we are right now in the aging field of pain, we are lacking a lot of knowledge. So even cross-sectional studies, if you would like to compare different age phases and have a group of young people or young animals versus a group of old people or elderly population and older animals, that would be really, really helpful. And I think we know that any knowledge that we could start generating would be really, really helpful for potential development of therapies, and not only development, but identifying and developing new targets for pain treatment.
Do you mind if I add one thing that occurred
Go
to me?
for it.
Please.
Yes, please
I
do.
was so inspired by this, and David, by the start of your presentation. And one of the things that so excites me about this attention to basic science is I have an idiopathic pain disease. It's actually my hope that in the course of doing the basic science around therapeutics, that you will identify things that allow you to halt or even reverse the underlying condition so that the therapeutics aren't necessary.
That's a great note to
That's the
end on.
goal.
David, go ahead. Yeah.
I'm going to say I think that's the goal that we all have. Yeah.
Absolutely. Yeah. Okay, thank you all. That was really terrific. Great presentation, David, and thank you all for a good discussion. And it's now time to move on to our next session, and we'll have Anne-Marie Malfait give that presentation. So this session is now disease-specific mechanism discovery for pain and disease modification. And we'll have Dr. Malfait, Dr. Markman, Dr. Shrivakar, and both Will and Christy as part of this one. So, Anne-Marie.
Thank you, Ted. So I trust you can see my screen.
Yes, looks great.
Okay, great. So I will actually talk about pain in osteoarthritis and a little bit about pain in other rheumatic diseases. So by way of introduction, I was actually trained in rheumatology, and I became very fast interested in the joint in general, and more specifically in cartilage, because at the time, I was interested in osteoarthritis, and it was very much believed that osteoarthritis is a disease of cartilage. So that's about 30, 40 years ago. But I then became later interested in the pain that comes with it for reasons that will become apparent very quickly. But basically, osteoarthritis is one of the leading causes of disability and chronic pain worldwide. It's the third most rapidly rising condition that is associated with pain and disability in the world, behind diabetes and dementia. And it affects primarily the knees, the hips, the hands, and the facet joints, the small joints in the spine. And so if you really look at it, it's such a tremendous source of pain. So for every 100 people who suffer from chronic pain, 30 of those, their source is osteoarthritis. 30 people suffer from chronic low back pain. And then there's also gout and rheumatoid arthritis. So really joints in general are an unbelievable source of chronic pain in the world. In the most recent epidemiological studies, it was shown that 600 million people in the world suffer from symptomatic osteoarthritis. And in the US, it's the third leading hospital discharge diagnosis, reflecting the huge prevalence of total joint arthroplasties, which is actually mainly knee replacements. And so recently also the HEAL initiative implemented a new initiative called Rejoin, because they identified this research need that most people with opioid use disorder in fact cite joint pain as a contributing factor in this. So I will just very briefly talk to you a little bit about what osteoarthritis is, because we're going to go back to why do we do basic studies to find targets for pain here. On the right, what you see is a joint at time of replacement with really bad osteoarthritis. You can see all the cartilage is gone. And on the left, what you see is an X-ray that's end-stage osteoarthritis. So what you can clearly see there is that there is joint space narrowing. That's because the cartilage is gone. And what you see is what people tend to call bone on bone. But what we now know is that there's all these different tissues in a joint participate in the pathology of osteoarthritis. The meniscus is involved, the fat pad is involved, the subchondral bone is involved, the synovium is involved, and all these tissues in the course of the disease participate and undergo pathological changes. Now, while we all accept that pain is something that really is in the brain, here too, I think there's such a tremendous opportunity to looking for peripheral mechanisms. And clinically, we know that there is very strong evidence that there is a peripheral drive for pain in osteoarthritis. And we know that for the knee specifically, because total knee replacement takes away the pain and also signs of sensitization in the vast majority of people, around 75 to 80%. So there is a very strong peripheral drive. And so therefore, I would propose that we really need to understand here what I highlighted in this red box to know all the different pathological events that are occurring in the course of this disease. The other point that I would like to make is I've shown you already this X-ray. Now, that is really end-stage osteoarthritis, and that is the result of biochemical, molecular, cellular processes that have been going on often for decades in people. And so we can really see, if we could go to the very beginnings, we can see very subtle changes in all these different tissues that I have already mentioned. And so the way this progresses in people can be then modified by mechanical factors like overuse, by hormonal factors, systemic factors, obesity, menopause, genetic factors. But then there's also the pain. And so when we look how the pain progresses in osteoarthritis, here too, we have this very long, sometimes decade-long process where people will have some pain, very often mechanically driven. Their knee will hurt a bit when they go up the stairs. Then that goes away. They'll have these episodes. They can have months where there is no pain. And as the disease progresses, this become more constant, persistent pain. The pain can radiate outside the joint. In very severe disease, there can also be flares at night. And all these people then also, what we now know due to large clinical studies is that people will also have changes that can be detected by quantitative sensory testing. So people will have sensitization, loss of proprioception, vibratory perception, and things like that. And again, all these different factors can now influence how this progresses, okay? So that's really the picture. So what I want to show is, if we're going to model this, that's why I said what I said 10 minutes ago, is that we have to be aware of this. And so if we look, because we are studying a different disease the whole time, so there's a milieu in the joint that changes the whole time. And when we look at animal models, and we routinely look for months on end in animal models, we see these longitudinal changes in pain behaviors in a mouse also. And so they can, for example, develop sensitization very, very early on. But the weight-bearing pain or changes in gait will only arrive 4 months into the disease in a mouse. So when we actually look at molecular changes in the dorsal root ganglia, there too we see that these are very time-dependent, and they are very different whether we look in early osteoarthritis or in late osteoarthritis when we study the dorsal root ganglia, for example. Another point I would like to show is that what we are now discovering both in people and in mice, that the actual innervation of the joint changes over time. And so we see that there is sprouting of nociceptors from the bone into the cartilage. Cartilage is normally aneural, and this correlates in people with pain. And we can also see this in animal models, in all different animal models of osteoarthritis. So one of the important points that I would like to say, that we really need to identify these neurons. We need to know exactly which neurons are they. And again, we've heard about topical treatment. The joint is actually beautiful because here we have an opportunity of going intra-articularly, and that's a way of also avoiding systemic side effects. So I just want to highlight here in one slide, one way we are addressing is this with rejoin, but this is really just the tip of the iceberg, and there are many people involved, is that we are collecting all these different knee tissues. We're also collecting innervating dorsal root ganglia and the spine tissues, and Dr. Price is involved in this, but many other people are involved. And we're really making a molecular profile of all the different tissues that are involved. So we're looking at the histopathology, we're looking at the detailed innervation. We're doing this over time in mice and in people. And so hopefully by then doing this in-depth transcriptomic profile, we can identify drivers of sensitization and neuroplasticity that are unique maybe to men or women, or to younger and older people. So I want to make two more points actually here, and these are, so when you look at this very long disease, and so I made this divider line, is that a very big question is, how are these related? How is that joint damage related to the pain development? And why is that important? Because in the field of osteoarthritis, we have this dogma that there is a discordance between the joint damage and the pain. And that is because most studies look at end-stage osteoarthritis. And so there is this radiographic osteoarthritis that people can have bad knees on a radiography and very little pain. Vice versa, people can have a lot of pain and knees where you don't see much on an X-ray. So we really need to understand this relationship, I think. Another very important point to make, and that actually also goes back to what we just heard from Dr. Julius on the endometriosis study, is what is the effect of the nerves themselves and the pain and analgesia on joint damage? So we know, for example, there is such a thing as an analgesic hip, where people on NSAIDs can develop accelerated osteoarthritis. And then sadly, just a couple of years ago, we had a very promising pain-killing agent, an antibody that neutralizes nerve growth factor. And the development of that was unfortunately stopped because a subset of people here developed rapidly progressive osteoarthritis. So the reason why I'm highlighting this is that we really need to understand that relationship. Then I have one last slide, if you don't mind, just to get away from osteoarthritis. Again, this shows in the United States, Europe, and Japan how prevalent osteoarthritis and chronic low back pain are, and how they actually eclipse other sources of chronic pain. But I would like to also say that joints are a tremendous source of pain, and there is so much that we do not know. And so osteoarthritis research focuses by and large on the knee, but the hip, there is still a subset of people that gets chronic pain, and non-inflammation need to be recognized. It's very painful disease, inflammatory low back pain. Nobody knows anything about where the pain comes from. Inflammatory myopathy, tendinopathy, connective tissue disorders such as Ehlers-Danlos syndrome, and juvenile inflammatory arthritis. These are all tremendously painful diseases where there is no research. And so that's the point that I would like to end with, that we really need to model all these diseases, and we really need to have tissues from these people with these diseases and really go look disease specifically what is going on there. And that's it. Thank you.
Thank you very much, Anne-Marie. So now I'm going to turn it over to Dr. Markman for his thoughts.
Well, I just thought that was a brilliant discussion of, frankly, of the complexity of osteoarthritic pain. And I'm frankly still meditating on your point about the challenge of modeling it, given the slow progression, and I think that really is something which we often gloss over, which is incredibly important. I also, I was thinking, frankly, about Will's point about understanding idiopathic syndromes. And the challenge of modeling syndromes that begin, ostensibly without a clear, form of injury or mechanistic cause, and you're, trying to describe that phenomenon. I think scientifically, from a modeling perspective, that seems to present such a challenge. And lastly, I just wanted to ask you, I think that Dr. Julius kindly put up a sort of a graphic which gave us a sense of where I think he thought were the important molecular targets that were to be sort of considered, as we think about peripheral modulation of pain intensity. I'm just interested in your perspective on what you would say are the sort of leading targets for, thinking about the changes in innervation that you described, and what mediates those. You alluded to, anti-nerve growth factor antibody, but are there others that are top of mind for you?
I wish I could say there are, but there aren't actually. To me, currently, I would not identify any. The main reason is that I think we are not there yet. I think we are only just starting to discover that there are that in pathological joints with osteoarthritis, there are nerves that sprout that should not be there. And although I showed you for one, these are very limited studies, and the ones that I showed you contain CGRP, but that's actually only just an example. There's very few. I really think we need to dig deeper. I don't think we're far off, but we need to dig deeper in really identifying the nerves that are mediating this pain. What I didn't get into is that there are nerves in the subchondral bone. There are also nerves in the synovium. This might be, they may actually have different identity. And so they may explain the different type of pain, the different types of pain that people may experience. So I think that we need to better profile the pain profiles in individuals, and then relate that with the type of target that they may have. So some, you know, in osteoarthritis, we talk a lot now about, you know, it's not one disease, it's all these different phenotypes. There are people who have more bone-driven phenotype, there are people who have more driven synovitis-driven phenotype. And so there are different types of nerves, and different targets may well be important for these. So I think that's the first step before we can go to what you just asked. The very simple answer for me is yes, it's still nerve growth factor. I think it's a really good target, by the way. We just need to understand why it caused in some people, in a very small group of people, rapidly progressive osteoarthritis. Again, I think it's an entirely solvable question that we need to solve by doing the right experiments, which I don't think have been done until now. Mm-hmm.
Just to follow up though, do you think that there's a sort of a linear or one-to-one relationship or correlation between the neurite outgrowth and the pain intensity? It seems to me that, you know, that anatomic feature, neuroanatomic feature in and of itself probably doesn't explain the complexity of the
Not
phenomenon.
at all. No, I was not implying that at all.
Okay.
No, I was just answering your targets. I think the targets will be neuronal. Another thing we haven't crossed on or touched on yet, just a little bit earlier, I think Dr. Julius was mentioned, is the crosstalk between the tissues. So there may well be targets in specific bone cells or in fibroblasts in the synovium that will regulate the nerves. But again, those need to be looked at, and those won't, in my opinion, be the same in all people. Mm-hmm.
You mentioned the disconnect between, you know, conventional radiographic imaging of the knee, for example, and pain intensity. And, you know, and do you think there are other imaging modalities, given the revolution in imaging that we're seeing, you know, MRI, bone imaging, obviously ultrasound? Do you think that, you know, those are...
Yes, that is well established. So I had to hurry through that. But so when you, the earlier you go, you can detect by MRI, for example, one of the strongest correlates to pain, weight-bearing pain, is a bone marrow lesion defined on an MRI in the subchondral bone. So what histologically, what that actually is less clear. These things also can go up and down. And so when they diminish, the pain goes down. When they come back, the pain, and that happens in the course of about 6 weeks. Synovitis on MRI also. So yes, I totally agree. If we could develop specific methods to go for live imaging, I would love to have live imaging of nerves in people to really see when and where they are active in a joint. And with weight bearing, for example, that would be a help. And again, I think that's probably attainable.
Just one last question. So akin to what Dr. Julius was describing, you know, with regard to looking across different diseases. I mean, do you think that this, for example, the subchondral T2 hyperintensity you see in a patient with osteoarthritis, this is in the knee or in a facet joint or at the end plate of a vertebral body, you know, or in a shoulder? I mean, do you think that we should be thinking of, you know, looking at organizing the sort of syndrome by looking at perhaps at that MRI feature and developing and exploring therapies for chronic musculoskeletal pain, or do you think that we should still be focused on OA of the knee and you know, OA of the DIP joint? I mean, because to me, I think, you know, a lot of what I've heard in both of your talks is there's a lot of, you know, common features to the underlying molecular neurobiology and other cell type biology that maybe prompts us to rethink how we're organizing, you know, sort of the way we structure not only the funding through the institutes, but also how we conceptualize these problems. And maybe we should be looking for therapies that, you know, take a feature like subchondral T signal change and develop a therapy for chronic musculoskeletal pain, as opposed to just, you know, thinking about the next treatment for intra-articular treatment of the knee. Do you have any thoughts?
I think that's, yes, I think that's a great point. And to me, an example for that is which is why I put it on the slide, we know nothing is spondyloarthritis. And so when people have inflammatory low back pain and they have changes in their sacroiliac joints, for example, you know, I recently went to a talk, lo and behold, they have these MRI lesions in the bone marrow, and that correlates with pain. But so this is exactly what you are saying, is that there might be that there's mechanism in RA. There's a lot of inflammatory pain. It's in the synovium. That might be different subsets of nerves, different specific cell types driving it. But you can have a more synovium-driven pain. You can have a more subchondral bone-driven pain. I think that's a very good point, and I actually that's exactly what I think is going on at this time. And there are methods to study that. So this is why I think so the disease-specific models where we can really look at that over time would be so useful.
Yeah, and I think to me that, again, I think this goes back to the point that, Christy and Will were making, to me is that, if we think about these features that are, important to the experience of pain in a broader way, ultimately, that may be a way to address the access to therapy problem, right? Because, if the therapy is developed in osteoarthritis in the knee, but we think the same process is at work in osteoarthritis of the distal interphalangeal joint or the facet joint, I think that I think what's a challenge is that, that benefit, which is probably there for underlying mechanisms across these different regional anatomies, frankly, can't be gleaned by patients. So I think that, how we conceptualize disease, chronic musculoskeletal pain versus a single joint, I think has importance at the front end in terms of designing our experiments and how we study them. But I think, frankly, it's enormously relevant for my own practice experience at the end of reaching patients so that they can have the benefit of these therapies and insights.
Very good
Thank
point.
you both. Yeah, so now I want to make sure that we hear from Dr. Shilvakar. So, yeah. So I know you've done some really amazing work on the brain. So it'd be good to hear your perspective on opportunities there.
Absolutely. And yeah, thanks for having me, Ted and John. So, Anne-Marie, I was really excited to hear you say, pain, we know pain is in the brain, but still fundamentally, we know it's also peripheral, right? So I guess just to introduce myself, I am an associate professor at UCSF. I'm a neurologist and a pain clinician as well, so I see patients, but I run a lab developing brain stimulation therapy or technology, let's say, for pain treatment, as well as trying to understand, basic mechanisms in the brain. And so, one thing that really I think inspired me, Anne-Marie, about what you're talking about is the potential nature of inflammatory molecular processes in knee osteoarthritis and how they might be different in, let's say, shoulder osteoarthritis or even like facet osteoarthritis. Clinically, it's interesting. We treat many of these the same, at least pharmacologically, right? We just say, oh, it's inflammatory and nociceptive. Let's try NSAIDs, anti-inflammatories. If it's affecting muscle, we'll target we'll try muscle relaxants. But what I do is I do device development and use devices as a potential therapy. And I know the HEAL initiative has been amazing in supporting new device development very early on, right? And so this takes a long time. We use devices right now to treat neuropathic pain, so nerve pain with nerve injuries. But it's clear that there's efficacy for inflammatory disorders and some nociplastic or nociceptive pain syndromes as well. And so I guess what would be really nice is to have folks studying mechanisms of, when devices work, specifically spinal cord stimulation or peripheral nerve stimulation, when they work, why do they work differently in these various disorders? I was fortunate enough to have one of these UH3 grants for technology development early on, and it's interesting. The focus has always been, I think, with the HEAL and NIH in developing technology that's commercializable and translatable. But there's also a separate, I'll say that's one end of the spectrum that HEAL's focused on. And we've been doing kind of invasive brain recordings and stimulation for various neuropathic pain syndromes. On the other spectrum, the science, or I'll say the technology in clinical treatment, the actual delivery of technology is way ahead of our scientific understanding, right? So there's another of how devices work or why electrical stimulation to nerves necessarily does what it does to relieve pain. And so on the other hand, there's a focus on studying FDA-approved mechanisms and explaining, all right, why do devices work? I would love to see something in the middle, or kind of bridging this gap, specifically understanding, okay, other mechanisms of actions or let's say ways to target in the brain non-invasively to stimulate, to relieve pain using TMS, which is approved and actually very commonly used, in Europe. Really hard to get access for patients here. I send my patients to a psychiatrist often with a diagnosis of major depressive disorder, one psychiatrist I know is has expertise in targeting the motor cortex for pain, and it's helped certain patients tremendously. So studying these, it feels iterative or it doesn't feel groundbreaking, but studying these relatively available non-invasive brain stimulation methods that are not FDA approved, but finding pathways to get them FDA approved, either with market or industry collaboration or otherwise, but really kind of taking technologies that are at the edge of approval in my mind and bringing them over over the line to make them available for patients would be tremendous. If Heal could somehow focus on this. I know that it's challenging because there's NIH Blueprint MedTech, right? It's like the neurotech hub, the almost the incubator. But sometimes physicians such as myself may not have expertise or even interest, let's say, for creating a company, or profiting off of this. We just want to see the technology brought to patients. And so it would be nice to be able to use the resources of Heal in some manner to bring these, again, liminal or therapies that are not quite there to bring them over the line. And so, yeah, that's what I'll say about that. And I really love that we're studying molecular mechanisms of fundamentally of diseases. And I think it would be great to study the mechanisms of therapy that we have that we don't quite understand, not just for FDA approved indications, which is currently, I think, the HEAL grants that are available, but actually for not necessarily approved indications, but understanding basic biology.
Thanks, Prasad. We have like 1 minute left before we go into the break. I thought maybe since we spent a lot of time talking about drugs, if maybe you could just talk really quickly about like what you see on the horizon from a device or tech perspective, that if you have an example of something that maybe is getting right to that point, like you were just talking about.
Yeah, so I think that I think TMS is a good example. Transcranial magnetic stimulation is a non-invasive therapy. It's already FDA approved for major depressive disorder in a different brain region, right? It's FDA approved for OCD in a different brain region. There's extensive studies dating like 40 to 50 years of TMS over motor cortex for chronic pain. I think the data is better for certain types of pain, like facial pain. And again, so it's interesting. I would say there's a lot of reticence from what I've heard, or I'm not even aware of all the obstacles, necessarily in bringing it to market for the indication of pain. But that's one example. Another example that I think is very exciting to even study as a possible therapeutic application, but also in basic science for pain, is low intensity focused ultrasound. Focused ultrasound it's not new, but it's being newly used in application for a whole host of neurological diseases. Basically involves multiple element transducers, that's basically penetrate the skull and create kind of an interference pattern of inhibition or stimulation. There's human trials going on. There's a recent paper in pain by Dr. Kubanek from Utah, and it's a therapy that holds a lot of promise. And I think exploring, fundamentally, how could we use this to actually treat pain, or even in a clinical trial to understand what patients might respond, what brain regions to target. There's a lot of basic questions. So those two technologies I think are very exciting.
Great, thank you very much. So everybody remember we have an hour at the end for open discussion, so we can come back to some of these points, but I'm going to keep us on time and we'll go ahead and go into our break because we've been going for more than an hour and a half now. So we'll take a 10-minute break and we will be back at I guess 11:50 Eastern time. So see everybody in 10 minutes. 11:50 here. Actually, I'm Central Time, so I've got 10:50 here, but I think it's time to restart. And for the next two panels, I'm going to hand it over to John Markman to be the MC. So, John, over to you.
All right, fantastic. So we're here in the second half today, and it's my privilege to introduce Dr. Salvemini, who's going to be talking about therapeutic discovery for pain. And then we'll have a panel after that, and that'll wrap up around 12:20. So Professor Salvemini, look forward to your thoughts.
Thank you very much, and thank you for having me here today. So I've always been interested in discovering non-addictive pain targets with the overarching goals of developing a novel treatment for the management of neuropathic pain states in particular. Before joining academia, I spent about 15 years in the private sector, where I was involved in leading drug discovery efforts on anti-inflammatories and analgesics. So in the lab, we use a multidisciplinary approach ranging from identifying and validating targets using non-biased approaches to then deciphering the molecular and cellular mechanisms engaged downstream of that particular target. And then what we do is that we use this basic science knowledge as our foundation to drive drug discovery efforts and identify lead molecules to move forward to preclinical and clinical developments using various mechanisms, such as, for example, the programs that are offered through the HEAL initiatives to the development of new startups or license-based partnership with industry. So we are particularly interested in G protein-coupled receptors and in particular the orphan GPCRs. So what I'd like to now do is really kind of share a couple of slides that may help exemplify our approach on the identification of these non-addictive pain medicine. So I am going to share the screen. Let me know if you can see it. Are we good?
I want to swap there you go.
There you go.
You good? Okay. All right. Okay, so most of you know that these GPCRs are probably the largest class of membrane proteins, and they represent the most druggable targets to make it to market. So there are about 826 human GPCRs, and about 35% of all FDA-approved drugs target GPCRs. However, there are a substantial number of GPCRs whose ligands are not known, and these are called the orphan GPCRs. And only a few FDA-approved drugs target these orphan GPCRs, and that's where our interest lies. And so over the years, we've identified several orphan GPCRs that are shown on this slide. And for the purpose of today's example, I will focus on GPR160, which is been a target that has been moved forward with a HEAL grant for target validation. So several years ago, we basically discovered that the GPR160 is an abundant orphan GPCR that is expressed and upregulated in spinal microglial cells in several models of neuropathic pain states in rodents. And the GPR160 is also expressed in human microglial cells in the dorsal horn of the spinal cord. This, on the left-hand side, you see data that is unpublished from Ted Price at UT Dallas and Diana Tavares Ferreira, where they use a single nuclei RNA-seq and detected abundant expression of this receptor in the microglia. And this is data from the Levine data set where, again, the target is found on microglial cells in humans. So what we then did was to use a number of genetic and pharmacological approaches. So we actually developed the first GPR160 knockout mouse. We use that mouse and we use sRNA approaches as well as immunopharmacological approaches to validate the role of this receptor in rodent models of neuropathic pain. and demonstrated that when you blocked that receptor, you prevented the development and the maintenance of neuropathic pain states. The ligand of this receptor was not known, and so what we did was to use a receptomic approach, which we published in this paper in the Journal of Clinical Investigation about 4 years ago, and identified cocaine and amphetamine regulated transcript, CARP, as a ligand for GPR160, and this data provided the initial deorphanization of the receptor. We then went further, and we started to decipher and validate the cellular and molecular mechanisms whereby activation of this GPCR signals in the spinal cord to drive neuropathic pain states. And this led to a couple of papers. A recent one is going to be published here in Pain. And an initial model that we put forward several years ago was that activation of the receptor by CAR peptides turn on the ERK and PrEP pathway to then drive the molecular underpinnings associated with neuropathic pain. So, up until now, this data was very compelling to us. Number one, because we identified the receptor and nothing was known about the receptor. We began to deorphanize it. We know that the receptor is present in human as well as in rodents. We demonstrated that if you can block it, you have a positive outcome in neuropathic pain states. And we began to understand the molecular mechanisms whereby activation of this receptor contributes to neuropathic pain states. However, there are no known small molecule ligands for this receptor. And so this kind of compromises our ability to understand the role of the receptor in pathophysiological settings and come up with potential novel treatments. So the data was exciting enough for us to decide to begin a small drug discovery effort to see whether we could identify small molecule hits for the receptor. And so what we did was to use a virtual screening approach paired with in vitro cell-based assays. This approach then led to the identification of a first hit small molecule GPR160 antagonist, which we hope to then build upon and develop in new compound. And I'm just going to show you briefly what we did here. We basically use a GPR160 homology model based on the structure of the P2Y14 receptor, and we docked in the CAR peptide onto that homology model to look at the potential to identify amino acids that could be critical for the binding and the signaling. And then we did a high throughput glide docking targeting specific GPR160 and CAR-P interaction. So this was about 2 million compounds. We then filtered these hits based upon the glide XP score and physical properties that nailed it down to about 20,000 compounds. Then the top compounds, we purchased them through various sources, and we screened them on the inhibition for adenylate cyclase in cyclic AMP assays. This is a GI alpha couple receptor. This led to the identification of about 12 compounds. And one of those compounds turned out to be pretty interesting as a hit. It has an IC50 in the low micromolar range. And we demonstrated that the antagonist can actually block the CAR-P mediated behavioral hypersensitivity as well as behavioral hypersensitivity in a model of neuropathic pain. So on the left-hand side of the panel, you see that when you inject CAR-T intrathecally, you have the development of severe mechanical allodynia, and we've published, and we can block this with siRNA against the receptor or with immunopharmacological approaches. But you can see here that these antagonists can block the development of allodynia to CAR-T in a dose-dependent manner. And on the right-hand side of the panel, you'll see that an intrathecal administration of the antagonist can reverse in a dose-dependent manner the development of allodynia following chronic constriction injury. This data then suggests that the compound blocks CAR-P GPR160 signaling. So where we are now, so the HEAL initiative grant for target validation was instrumental for us to target validate the receptor and provide exciting data that allowed us to then identify a small molecule through our own funding. And what we are now in the process of doing is to isolate and purify the receptor and then develop high throughput screening for drug discovery. And these experiments are in progress, and we'll be looking for funding. And we hope to identify lead molecules then to move forward through preclinical development through various opportunities, including the wonderful HEAL-funded programs that are available. So, you know, I think that this has been hopefully will be a successful example of what can be done with basic science discovery and moving those discoveries to hopefully in the clinic. As Dr. Price started off our meeting saying that, you know, non-addictive pain development has been a cornerstone of the HEAL initiative for several years, and significant progress has been made in this space. I personally feel that continuing these programs is really critical, as they provide investigators such as myself and many others with the tools that are necessary to translate our innovative ideas from the bench to the bedside, potentially transforming pain care while increasing our fundamental knowledge underpinning pain. I also feel that continuing to put resources behind these programs becomes even of a more pressing conversation, considering the decline in industry-wide pain therapeutic development. I strongly believe that non-addictive pain development programs are poised to accelerate innovation, to increase basic science-driven translational research, and increase the commercialization of new products that will hopefully help millions of patients in need. Thank you.
Thank you, Dr. Salvemini. That was excellent. I'd love to hear from Dr. Price, if you want to start with your comments.
Yeah, sure. That was really fantastic, Dr. Salvemini. Thank you so much. I guess I'll just echo what you said in your closing, that I think the HEAL initiative and the opportunities for drug development projects, in particular the UG3, UH3 projects, of which I've been able to be part of two, have been totally transformative for the kind of work that we do. So, you know, the first UG3, UH3 that my group received was for development of MINC inhibitors for the treatment of neuropathic pain. And this was something that my lab had worked on for almost a decade before we started doing the drug development project. And we applied right when the first UG3, UH3 calls came out from the HEAL initiative, and we were in the first cohort. And we've been lucky enough to go all the way through from identifying a lead candidate through the IND enabling studies. And now we have HEAL support for the phase 1 trial that we're currently doing. So it's been an amazing process for us. And I also just want to point out the incredible support that we've had from NIH throughout the whole process. Obviously, the funding makes a huge difference, but on these projects is really not just the funding that enables the work to go forward. We had an entire group led by Anne-Marie Pellimonter, who I saw was on earlier, but I think she's not here anymore. But you know, she and her group of advisors at NIH played an absolutely amazing role in helping us to make good decisions about all of the work that we needed to do to get through the IND enabling process. And we also had access and support from contractors that NIH had put in place. And I know many of my other colleagues have had the same experience. So I just want to emphasize how incredibly well put together this program has been, how it's enabled us to go from basic discoveries, like Dr. Julius talked about, all the way through to having a drug that is now getting really close to hopefully going into efficacy studies after we complete the phase 1 safety. So I'll close there, John, and turn it back over to you.
Thank you, Ted. I'm just interested to hear, is Todd on the line? I don't see him here. I'm not sure if he's with us today.
He was earlier.
I think Todd had to got pulled away for something and had to leave, unfortunately.
Okay, great. Christy, I don't know if did you want to comment on this?
You know, I'll be honest, a lot of what's being talked about is a little bit over my head. But I've been trying to follow along, and one thing that actually crossed my mind while I was watching the presentation was, you know, allodynia, which I have in my hand. And, you know, when you were talking about, you know, it reversing some of it, and I assumed it was the same thing. Again, I am not trained in the areas that you guys are, but I am become somewhat familiar with the things that impact me. And I know that the allodynia I have is along the median nerve, and so is a lot of the other neuropathic pain symptoms that I have. And I guess that was the only question that kind of popped in my head was, you know, is there any reason to believe that it would also address like the autonomic and the pressure changes and things like that that would run along those same nerve pathways? You know, the treatment and the study that you just talked about with the mice. But, you know, in terms of commenting about the science, you know, you guys are, it's fascinating watching this stuff, but I don't have the bandwidth to offer anything in that regard.
Well, I think that's actually a very incisive question about, you know, which features of the neuropathic pain syndrome, you know, are being modeled and which ones are not being modeled. So Daniela, do you want to comment on that?
Well, I mean, so the studies that I have just shown you are all with, you know, evoked pain behavior. We are starting with this particular project. It's been a little bit difficult because there are no small molecule ligand to go in and actually block the receptor. So we're going to start now studies looking at non-evoked pain behavior with, you know, the molecules that we are going to be developing. So we'll add that other dimension to the work to get a better understanding on the profile of action of these, you know, antagonists.
Well, good. I'd like to open it up for others too, but Ted, did you want to add or?
Yeah, maybe I can just comment on that too. I think we've taken a slightly different approach in our group, and that is we developed this collaboration with the Southwest Transplant Alliance, which is a organ procurement organization that's very large and based in Dallas. And what we've been trying to do is to work with them to recover dorsal root ganglion from organ donors, and we now get pretty extensive medical histories. And while we obviously can't study behavioral aspects of pain related to the mechanisms that we're looking at in dorsal root ganglion neurons that we do recoveries from organ donors on, we can look at disease-related mechanisms. So, for instance, for our MINC program, one of the things that we developed there was to look and see if we could see engagement of the signaling pathway in human nociceptors in people that died and became an organ donor, but had a very clear history of diabetic neuropathic pain. And there we saw very clear engagement of the pathway, which really was a huge finding for us that helped us to move forward in getting the funding that was needed to do the phase 1 clinical trials. So there can sometimes be this big gap in your ability to make the jump from the preclinical studies to the clinical studies, because the amount of money that's needed to do the clinical studies is you know, it's a very substantial investment. So that's been a huge thing for us, is being able to develop the more kind of human molecular rationale for developing these targets.
Great. Would others from the committee like to comment or make a point here? Well, one question I have is, you know, I think from Dr. Malfay's presentation earlier in the day, you know, thinking about the time course of the conditions and the relationship to the animal modeling. And I'm just interested here, do you think there's an analog too in in the neuropathic pain world as well, given that, you know, these are often relapsing, remitting conditions with or very delayed onset in terms of when the nerve injury is and how that relates to the presence or absence or severity of pain intensity. Dr. Salvemini, so I'm just interested in the challenges of modeling. Does you think this translates across all, you know, syndromes, whether it's neuropathic or nociceptive or inflammatory and arthritis, or do you think that those are distinct, and in what ways are they different, or if you can contrast that or the same?
So if we speak, I mean, if we're talking about this specific target, well, so first of all, you know, the target is probably going to be different amongst the type of pain states. Sure. And so whenever we think about developing a, you know, non-addictive therapeutic for, say, neuropathic pain, we definitely want to, and we have profiled it across several models. And not only one, with the idea of potentially honing in the best area that these kind of, you know, therapeutics could be important in. So definitely there's going to be differences in the response to the drug, and we've seen differences in the response to various therapeutics targeting these, in our case, orphan GPCRs across various models. But the information that we got out of that was very important, because it allowed us to say, okay, you know, mixed inflammatory neuropathic conditions may not be appropriate, whereas maybe, you know, pain caused by chemotherapy is a better target, and so forth. So we don't stay just with one system, but profile many.
I certainly see the value of this sort of cross-validation through different mechanisms of onset. I think, you know, obviously one challenge, which has, you know, been, you know, famously difficult for the field, is using these models to decide which phase 2A population to enter, and feeling though you've truly tested the hypothesis that these mechanisms are relevant.
Right.
You know, you to test it in just osteoarthritis or just chemotherapy-induced peripheral neuropathy, you know, there's often the sense that you've missed out on the opportunity to explore the full potential of a target or a mechanism before discarding it or before moving on to another. And so I think this is, I think, one very important challenge. You know, there are, you know, I think that there are multiple platform efforts, both in industry here at Eli Lilly, but also platform efforts at the NIH through the EpicNet, I think, which are trying to address this bottleneck at phase 2A to try and help us triage these decisions and decide which patient populations, you know, must we test, how should we approach it, and how can we do this efficiently. So we know, because I think there was a great point earlier by Dr. Julius about perseverance, you know, and how this takes a long time for us to sort of work through the mechanisms that are relevant to human disease, whether it's in cystic fibrosis or, you know, in understanding cardiovascular disease. And so I think that there's one of the challenges here is, you know, the question of, you know, how many times, you know, to try in phase 2A to sort of replicate what's been hypothesized and demonstrated in an animal model per se, versus when to sort of say that we've fully interrogated that hypothesis and it's time to move on. And so I think that the efficiency of that step is a real challenge, I think, and I'd be very interested to hear your thoughts on that and others on the panel.
No, that's a great point, and I think that in this respect, I think that you know, the HEAL initiative. The HEAL initiative for the preclinical screening platform for pain that has been developed to accelerate the discovery of non-addictive therapeutic has been really, really important, because, you know, it provides investigator the opportunity to look at their target molecule and profile it across various models that perhaps the investigator does not have in their own laboratory. So I think that that initiative has been really kind of tremendously important in what we are discussing here today. So
that is a great point and very relevant to this.
I was going to mention that there's a comment in the chat about autonomic symptoms, and I think that's an important aspect of this as well that is sometimes overlooked, and taking a kind of more holistic approach to what's observed in some of the animal models, and also a better understanding the complexity of the experience that the patients have can help us to hone in on some of these other things that are very common among people who have small fiber neuropathy and neuropathic pain.
Other comments or thoughts from other members of the panel?
I was just going to say that I think sometimes, people sort of lose faith in this mechanism-based approach, sort of what John was saying. I mean, it is hard to know, especially in drug development. It takes a lot of money and you have to make tough decisions about what to pursue. But I think, I think we still have to keep our eye on the ball. In modern biology, I think the only real way forward, you can get lucky by throwing darts at the wall, but obviously, you have to understand mechanism. And I think, my example from the previous, for my presentation, the first slide was really, part of it was to show that even when there was a good target, sometimes the first few drugs failed for various reasons. And if you think it's a really good target, then, it's worth coming back to at least at some point, and possibly in the setting of, different types of pain syndromes. But the more we know about these molecules and how they work at the structural and biophysical level, the greater the possibilities that we can make drugs that either will be more efficacious or will modulate these receptors and channels in ways that don't have some of the side effects that have beset early, drug development trials with these targets. So, you can't follow everything, and sometimes you can't beat a dead horse, but But if we think that a target's really working for something, it might be particularly efficacious for one application or another. I think it's a mistake just to sort of move on and put that stuff aside and just say that approach or that target's not going to work. I think we have to keep an open mind and come back to it when we think there's either a new insight into how that molecule can be regulated or a new setting in which modulation of that molecule or pathway might be particularly efficacious. And I say that because I'm concerned that there's been some loss of faith in that in the pain area, and people are thinking about more complex things that, where it's going to be really hard to evaluate how molecules or combinations of drugs work, et cetera. And I do think we have to have faith in the long-term process of keeping our eye on the ball in terms of really understanding mechanism. I think that's the only way forward in terms of really taking a rational approach to this problem.
I think that's such an interesting point, sort of the sort of rational approach forward drug development, sort of to me, the inductive approach. I do think it's also interesting, when you look at the history of analgesic development over the last half century or more, much of it has really been, through serendipitous clinical observation in specific patients and taking the hypothesis generation, which comes from those observations, and really using that to drive, I think, some of the mechanistic understanding and sort of back translating. And so I'm interested in the perspective of the panel on that idea, because, I think one of the challenges as a clinician, I think we face in 2024, which is only going to accelerate, is that it's going to be harder and harder to sort of take the phenotype in front of you in a patient and hear their specifics of their story and work backward, for example, in the way that was done with gabapentin or gabapentinoids, seeing the benefit from an antiepileptic and back translating it into understanding, calcium channel modulation mechanism for pain. I think that, as our clinical encounters get more rote and more templated and frankly less human in all of their dimensions, I think the ability to have a spontaneous observation is eroded. And so I'm interested in sort of the scientists here and their perspective on whether that's an important concern, and if there is important concern, what are ways to mitigate it.
Maybe I can make a quick comment on that. So, I mean, I think it all kind of comes back to where we started again with the importance of the basic science. So, not all basic science is necessarily hypothesis driven. Sometimes just doing really high quality work to understand how things work and having a keen sense of observation can lead to really important insight. So I think, we've shifted a lot of what we do to working with human nervous system samples, and part of the reason for that is because we have the opportunity to do so here, based on the relationship we have with the Transplant Alliance. But we've gotten back to doing very basic things, and we on occasion have made some observations that have really driven us down routes of investigation that I never dreamed that we would be working on. So one instance is this kind of rediscovery of these Nugget nodules that Stephanie Shires in my lab has made, where we see this incredible degeneration of subsets of neurons in the DRG of people with diabetic neuropathy. And I can't tell you that I know exactly what it means yet, but certainly has us thinking differently about the disease. And a lot of what we are rethinking fits very nicely with the clinical observations that haven't always been very well understood. So I'm pretty hopeful that that is going to spur some new areas of investigation and some rethinking about how to generate models that will better help us to understand what's ultimately happening in individuals that have diabetic neuropathy.
Very good.
Mhm.
I'm also just thinking about all the promising, targets in pain now that I see have been triaged from other areas of medicine because there was a spontaneous observation of pain relief in a patient who had chemotherapy or some other treatment, and it wasn't anticipated, and sort of that has led to a new mechanistic hypothesis because treating another problem, it was identified that there was pain relief. And I think that's that's a recurring story also. So I think and again, I think that's a harder thing to systematically fund prospectively. But I guess, I think one struggle is, how to create an environment where those sorts of observations can be fed back into the mechanistic research in order to sort of, what I would think is to sort of turn the flywheel of progress. Because I think that the key thing here is to make sure that there's sort of this bidirectional sort of, effect of where the hypotheses are coming from. Especially a lot of times when we sort of see an observation in another clinical area, but then it requires going back to the basic mechanism to refine the drug mechanism of action, to figure out the dose, to figure out the tissue compartment of interest, to figure out the PD relationship, right? So, even if the spark comes from another area, in order to repurpose it or to sort of fashion it as an analgesic, it requires so much understanding of compartment, tissue penetration, kinetics, to do that. And so I think that's one area of challenge, and I think it's one area where we could think about how can the HEAL initiative in the future play a role in accelerating, that path of discovery and development as well.
Yeah, in particular for repurposing drugs, and that's an area that is under considered, in my opinion. Fast track mechanism for repurposing drugs.
No, I think the acute observations of clinicians is unbelievably valuable, and that pertains to genetic observations, identifying rare mutations in genes, and it also what you say in terms of one flip example would be the original development of 5-HT3 antagonists for pain didn't turn out to be very effective for that, but then serendipitously turned out to be found to be a really fantastic antiemetic. And then, research going forward, trying to understand the mechanism of that by looking at expression of those receptors in places like the solitary tract, nucleus of the solitary tract. And so, we hope the same will be true for drugs that were used for other things and then found out to be, useful for pain. And yeah, once an observation like that is made, a clinical observation, then, it's really important to figure out what the underlying mechanism is. So to reiterate that, I think there's no substitute for really astute clinical observation.
Well, great. I think we're heading into thank you very much for that, David. I think we're heading into our last session now. So it's a privilege to introduce our last speaker for our last module here. It's Dr. Moreno. So Anna, thank you so much for taking the time to speak today.
Thank you so much. It's really an honor to be here. I wanted to just give my background as how I stumbled across pain. I did my PhD in bioengineering at the University of California, San Diego, and my PhD was really focused on development of gene therapy platforms, specifically for the epigenetics. So the ability to up and downregulate genes of interest without nicking or editing the genome, and specifically around CRISPR dead Cas9 systems. I tell this story often, but it's what happened. I was a board grad student on a Sunday night, had nothing to do. So I was reading papers and I came across the NAB 1.7 story about how, patients that have this loss of function don't feel any pain. And that's kind of what clicked in my head. You know, I was developing tools to be able to modulate gene expression. Why not go after NaV1.7 for chronic pain? Thankfully, I had a really supportive PI that was really open-minded in terms of entrepreneurship, Prashant Mali, who came from George Church Lab at Harvard. And so that's how this whole journey started for me. Once we started demonstrating proof of concept in mice, not just the ability to reverse pain, but also prevent pain, I didn't want to just move on to the next project. I really wanted to bring it to patients. So that's what started my journey back in 2018 is when I incorporated the company, applied for my SBIR grants. I remember one of the comments back was, Ms. Moreno doesn't have a PhD yet. So I was still, you know, not even graduated. But we were able to receive 2 SBIR grants, which allowed me to go from PhD to startup. That meant that I went from, you know, being a grad student to leading a company at J Labs, an incubator at Johnson & Johnson. So I wanted to focus today on my experience with starting a company focused on pain and what can be done, or, you know, thoughts around how to help us translate some basic science to really bring these therapies forward, and what are the challenges along the way. And so starting with the current state of private investment in pain, I'm sure some of you have seen this already last year published in Bio 2023, in which we see that the prevalence of pain is one of the highest, as we know, in the US, as compared to the amount of venture funding, the amount of financing going into it. So you can see a huge discrepancy in terms of cancer funding versus pain. And pain management in general needs to outpace investment, right? Where, again, the amount of money it costs the US to treat patients is higher in pain and addiction and other disorders, but lagging in investment. And this slide also just shows that there's 41 times more funding for oncology than there is for pain. You can see a lack of growth in pain oncology. So around the years here, looking at the increase in investment here in cancer, you see a nice curve of increased investment, and pain is just pretty much flat. And specifically, for example, most years, pain gets around $200 million in investment, and in which, you know, for example, oncology is 10 billion. So huge discrepancy there. Most of it has been focused on reformulation. So I'll talk about, you know, investments and risks, how do investors think about investing in pain. And so a lot of them like mechanisms of actions that have already been proven in human to just reformulate and invest in those drugs to maybe have fewer side effects or have, you know, different PK/PD. And, you know, on average, only 11 companies have been funded as compared to 109 companies in oncology year over year. And so I would say that obviously the biggest issues for challenge for financing, like investors, right? My experience when I was really excited about starting this therapeutic company and bring it to patients, and I started pitching it to investors. And the biggest feedback I was getting was, oh, placebo effects are really large. I've invested in pain in the past. My phase 3 drug failed. I lost a lot of money in pain. Do you have any biomarkers, or how can I know that you're actually hitting your target, or that you're actually seeing that decrease in pain that's not just patient, you know, the 1 to 10 scale and different scales, right? So that's the huge development risk that these people see. And specifically, for example, it was also noted that 0.7% of drugs make it in pain, whereas compared to 6% of other drugs are eventually approved. There's also regulatory barriers, as we know, following the opioid epidemic. It's been more challenging with the FDA. Something that I've learned with small molecules specifically, barriers at the FDA level that they might want more, you know, endpoints, more additional information, additional data to actually get towards drug approval. So that also creates some issues. And lastly, market uncertainty. Obviously, we can't really focus on this ourselves here at this conversation today, but we've seen, for example, a huge decrease in investment in the past 2 years based on interest rates and on inflation. And so market uncertainty definitely has a big impact, specifically on things such as pain. I would give some positives. So on the positive side, we have seen, and we're excited to see that Vertex is pushing along a NaV1.8 small molecule. And I've seen actually a lot more investors opening up for conversations because of what Vertex has seen. So it goes in waves in terms of pain investment. When I started, it was really down in the dumps, and I'm starting to see now more interest in investing in pain. And that's because of that human proof of concept that Vertex has. So that's another point I would make here. thanks to HEAL, we're seeing obviously an increase in investment. I started the company right at the point of, being lucky. Really, sometimes it happens when we're doing investigating, when we're trying to develop drugs. So we were able to get multiple grants from the HEAL initiative. But obviously, we need to bridge the gap, right? The HEAL cannot cover a lot of these fairly expensive costs. So what should we focus on? What can we, what would bring the biggest value? What would decrease the risks for investors or pharma as well, right? Because it's not just investors, pharma also have certain risk adversary. And I would say 50% of drugs are actually discovered and developed in biotech, and people don't know that. It's like 50% of drugs, big percentage of drugs are actually developed by small companies. And so that's why this is such an important area for us here. And so what are the challenges for a small company working on pain? I would say, one thing that I would mention is limited in-house expertise in key development areas. Obviously, as compared to large pharma, small companies don't have the resources, both in people and money, to really build out every single area that's necessary for drug development. There's also need for translational science support as well. And then difficulty in attracting investment and partnerships with early stage assets. So I'm going to try to talk about these points and how I'm thinking, and obviously at the end of this, we'll have a panel of other PIs that have also co-founded pain companies, and how we're thinking what can be done to help companies working on this. And so the first area here is limited in-house expertise. Like I mentioned, for example, my company right now is a team of 10 people that are in the lab, but we also have advisors and mentors as part of the team. However, there's always need, right, for additional expertise that might not be a full-time position, for example, or something you can't afford at that moment. And so one thing that I think the HEAL has done an excellent job on is some of these programs like the UG3, UH3, the collaborated efforts. We were lucky enough to receive a U44 grant. And so it allowed us to have access to some of these experts at the NIH. But the thought here is, instead of funding a 5-year grant that requires, multiple years, what if it's more of a smaller projects like the diversity supplement, where you apply, you have a certain amount of hours that you need, and that's it for companies that are either HEAL funded or applying for HEAL. Another idea here would be, for example, on demand office hours. So again, around the areas of CMC, regulatory expertise that might not require a full-time position. Something that we've mentioned here is guidance for clinical protocol development and IND. So pain is, obviously, clinical trial design around pain is difficult. There's placebo effects. There's other aspects of lack of biomarkers. So how can we better think about designing protocols that maybe have worked for others, taking others that gone through in the past, or ways that we can potentially even think about developing models, right, computational models to do patient stratification, or potential efficacy requirements of different drugs. And I wanted to give a shout out here to Emily Cabrera on the last point here, dedicating a supplement for key hires. So there was this call that for HEAL funded companies to enable us to hire people that had business development, finance, commercialization experience. Because another thing that, going from a startup to from PhD to startup, it's a team. And the fact is that people want to see people on your team that have that business development expertise. And so that allowed us to hire a chief business officer that has then pushed a lot of these partnerships and venture financing conversations that have really helped the company. So I do think this is important, just to think about, just again, helping us push along these therapeutics for patients. On the next point here, I wanted to mention translational science support. And so again, there's resource limitations in small companies. So some things that are difficult to fund that actually drive or de-risk the opportunity for investors or pharma, and that I've had experience with is, for example, larger animal tox studies that are obviously very expensive. We were able to take, one of our grants paid a portion of our large animal studies, as did some of our personal investment. But that really has opened up a lot more interest in terms of investor interest as well as pharma, showing that it's safe. CMC is important as well. And I'll mention the one last point for here in this slide, the NIH core services. So we were lucky enough, as someone mentioned earlier, the NCATS has a grant for small molecule screening, but they also have this iPSC derived collaboration. So we have a collaboration with the NCATs where we send them our gene therapy lead candidates and they test it in iPSC-derived nociceptors for us. So it really allows us to have nice external validation beyond the company that our drugs work, as well as bring in additional expertise that might not bring the capital into the company, but allows a lot of saving of capital outside. And so one thing mentioning here, for example, Ted Price mentioned earlier, the access to primary tissue, I think is also difficult for companies. So we've been working for a few companies here and there, and we sporadically get human tissue. But if it's something that we can implement potentially at the NIH, but we can have a collaboration or some kind of network to allow us to have access, not just small companies, but other academics that might not have access to that in their institutions, I think that will be really helpful throughout. And the last area I would like to mention is clinical trial support. I know this is a wish list and I know it's difficult, but a lot of feedback, not just from my company, from other friends of ours that are also working on pain, is a lot of, of these venture or pharma wants that phase 2A data, that proof of concept. And I want to focus specifically here on novel pain modalities, novel pain targets or modalities. We've seen NaV1.8, Vertex, we're really excited about that, like I mentioned. We're seeing multiple companies being funded for NaV1.8 because there's that human proof of concept, right? So now hundreds of millions of dollars are going to companies that might have been struggling along the way because there's that human validation. So if we can focus on validating novel targets in human that have never been tested, I think it's going to open up for multiple different modalities, potentially in that pathway or in that specific target of interest. Another aspect here is dedicated phase 2 funding. I know it's difficult, but potentially having some kind of public-private co-funding partnership where potentially the NIH can help co-fund part of this effort, and then private investors or larger pharma companies, when there's early positive data, can then unlock additional funds from private sources. So these are some of the thoughts I had along this area. The last point I'll make is that we've had, we've been trying to, and always talking to patient advocacy groups and patients themselves, interviewing patients, because we want to understand what's important to them. And so thanks so much for ensuring that there's patients here today, but I think that will be also really helpful if there's surveys or if there's a way for us to have more conversations with patients in a centralized fashion. So thank you, and looking forward to the discussion.
Anna, that was a tour de force, so very succinct and really compelling. I want to give Daniela and Ted a chance to speak to that. I know they have both so much experience in traversing these kind of waters. So Daniela, do you want to start with your thoughts on
so thank you very much, Anna. That was a really a tour de force. So I couldn't agree with you more that getting more help in the early stage of the process from the HEAL initiative to help de-risking the targets is going to be very, very important. There's a lot of reluctancy from VCs and in industry to put funding unless the target is being de-risked and less until you are, near an IND enabling studies. The HEAL initiative, there are programs that, like the U3, that has helped in providing guidance for CMC support and talks and so forth. And maybe we want to see more of that. So I agree with your concerns and share what you've been suggesting on how to perhaps increase the development of small molecule or whatever molecule into phase 1 and de-risk it. I also feel that we should have more emphasis on how to push forward drugs that are already FDA approved for other indications. And from my perspective, I think we're lacking behind that. And I've experienced that firsthand with drugs that we feel needs to be repurposed and tested as novel mechanisms in pain, where there is substantial preclinical data and also clinical data regarding biomarkers, and have been enabled to push them forward for a proof of principle in the clinic. So if we see more of that, that'd be great, because we're sitting on targets that are very promising and that have a lot of potential and years of data behind it.
Very important. Ted?
Anna did such a great job of going through all of the many issues, many of which I've also experienced. I don't have a ton to say. I'll just maybe highlight two things. I think the idea that Anna proposed of a public-private partnership is a really good one. I've heard this come up many times, and in relation to pain, I haven't actually seen it materialize, but I continue to think it's a really good idea, because it is very challenging, as Anna described in detail, to raise the money at early stages, in particular for targets that don't already have pretty extensive human validation. And pain is viewed, neuroscience across the board, I think, is viewed as being very risky, but there's obviously a huge medical need. So I think that's very important. And the other thing I'll say is that I think that there are a lot of really great ideas that are ripe for going out of universities and into small business innovation that don't happen because there aren't more people like Anna that have the guts to take it out of the university and make it go. So the reason I became entrepreneurial was not because I had the guts to do it. It was because I encountered some amazing graduate students that had a good training in entrepreneurship and finance, and also had a huge desire to do this kind of thing. And I've just been super lucky to be able to work with these people. So I think that we don't do enough in our PhD programs in some cases to support PhD students that are very interested in learning about entrepreneurship and learning about finance. I think it's very challenging for people that don't have a background in science to come in and lead these companies at early stages. And the other thing that often happens is that venture capital firms will decide to put some very senior person in charge of it who might work 25% on it. And my personal view is that that's often where very good projects go to die. So you need passionate people that really believe in the mission to make this happen. And I don't feel like we do enough to support that right now. So I think those two areas are places where the HEAL initiative could potentially make a really big impact.
I'll bring up the
Yes.
diversity supplement that does try to help, undergrads, MS, graduate students from diverse backgrounds in pain to help them become entrepreneurial. But I agree. I mean, for me, it was more of a figure it out versus, any kind of training along the way. I obviously took some business classes with MBA students, but yeah, I mean, potentially even having more panels where some of entrepreneurs come in and provide help with people that are wanting to, academics that are wanting to start companies, I think might be a way to help some of the things we were talking about today kind of come into the clinic.
Yeah, I think also what struck me on about your talk was, there's this, your point about what are the sort of in-kind or other forms of support which one can marshal, certain types of expertise or running certain types of experiments or regulatory support or trial design support, because I think that, discharging the different tranches of risk is challenging, right? And I think that, obviously for one small organization with 10 people to discharge all that risk to get to the later stages is almost insurmountable, right? So one thing I think is evolving, and I've seen this a little bit in this role that I've taken, is I do think there are ways in the ecosystem now to sort of offload some of that risk, through different relationships, at different stages, whether it's for a particular set of studies in toxicology that you need, or a specific set of regulatory runway that you need. And I'm interested to hear from you all whether you think that's been useful and important, because they're just ways to sort of interdigitate public sector funding with private sector funding, not necessarily in a wholesale M&A type, mergers and acquisition type way, or asset purchase type way, or licensing, but maybe in a more, I think, sort of either task specific or risk discharge specific way. So I'm interested in your views as entrepreneurs, whether you think that's useful, and if you think there are ways that the NIH could help sort of, sort of bridge the gaps or create mechanisms to better sort of align, the suitors with the risk reduction mechanisms.
So if I understand correctly, John, your thoughts is not so much in terms of how a pharma partner can come in early and help the risk. You mentioned more like certain milestones that would propel investment or a potential partnership and what the HEAL can do.
Yes, or just it seems to me that like there are certain regulatory issues which a partner can resolve for you or help you handle, which may otherwise divert all your attention and resources to that. And I guess so to me there's whether it's a certain set of toxicology studies or a certain set of assays or again there are many places along the value chain where I think that there may be ability to partner that's short of de-risking the entire opportunity or the entire enterprise.
Yeah, it's a really good point. I think for us, it's been a balance, right, of what we think the FDA is going to need, or like the conversation with FDA versus what investors want to see as well. It's also a challenge because, for example, sometimes we don't think we might need a large animal study, but that's what they want to see, right? So it's also that balance of finding those things. And for us, we have had opportunities to get an interest from pharma. And they have offered some of these, for example, okay, we can have conversations around your regulatory needs, which is great, right? Because they're interested in the opportunity. It might be too early for an M&A, it might be too early for a collaboration, but they want to make sure and they want to see the drug succeed. So they're offering some of this feedback and advice along the way. So it does happen, and it's great to see. I would also mention though that it's also been a challenge in terms of pharma coming in and out of pain. So we see people coming in, some of the large pharma all in, and then next year they're out. So it's also that challenge of consistency. There are certain, like Lilly, for example, is very consistent with staying in the pain space, but a lot of them are not. But some things that I mentioned in the slides are, for example, these very targeted, grant mechanisms that, okay, you only need this to get to IND, like there's just CMC, just the tox study, right? And to Daniela's point, in terms of CMC may have been de-risked for small molecules, but we're also dealing with novel modalities for pain. We're doing gene therapy, it's AAV, there's antibodies, there's other, RNA target modalities. And so those are also different challenges because gene therapy itself is challenging, and then add on to that pain, right? So you get this double whammy, but for the same reason that we think it's going to be successful because it's a different modality.
Yeah, I mean, so building on what John and Anna you're saying, I mean, all of this expertise in terms of CMC and tox and whatnot comes through funding mechanisms. So you've got to go in and submit a grant, and if you're lucky, whatnot, you're going to get it. So you're looking at very long timelines anyway, between the time you submit, between the time it's triage and you go back in. So these are your U3s, G3s, where clearly, they provide expert, advice for toxicology and CMC and things like that. But if we're thinking about maybe shortening the time of de-risking and potential partnership, then one could think about something along what the HEAL initiative has done with the preclinical screening platform, right? For pain, where there is a, really nice funnel that, investigators or small businesses take their molecules and send them over, and then it kind of dilutes out and dissipates. And at the end of the day, the investigator has, really compelling data of their molecules in various things. So maybe one could think about a similar strategy whereby HEAL sets up something similar to that, where you have, where you provide expertise to PIs or small businesses on a real-time basis, kind of. You don't have to wait for a grant, of CMC experts, of toxicology, of A, B, and C, right? And that could be a way of expediting and increasing knowledge, because you're going to have access to experts for the CMC for your particular molecules, toxicology for your particular molecule, PK/PD for your particular targets, without having to wait, a year, 2, 3 before going through the grant system. So I think that is why, I think the preclinical screening platform has been very successful in really helping investigators, both in industry as well as in academia, to propel their discoveries on target and various animal models. So that's just my thinking around that, because there are mechanisms that we have right now, but they're very, very long. And until we de-risk, we're not going to get money from anyone, more or less. And the other thing I think that what you mentioned was very important was the fact of having accessibility to human tissues. One of the things that people want to know all the time, is a target present in humans? Of course, we need to have the target present in humans, right? Because you're not going to do anything if the target is not in human. But then how are you what experiment are you going to show, like what Ted was talking before, about showing that, the target engaged in that particular setting is, the similar in rodents to humans. And, there's only a few centers that have access to all of that. We don't have a lot of places where they have access to a lot of patient samples, disease, diabetes, cancer, and whatnot. So that is also, I think, very important in the equation of finding out how can we expedite that component and into the equation.
I agree with that. And I think, one thing Anna and I have talked about, is really, I think, something to think about. When I was struck by your comparison to oncology investment to analgesic development investment. And obviously, one key difference to Daniela's point is that the human validation in oncology comes much earlier in the drug development process, because, phase 1B is often done in patients with, life-limiting illness. And so, one thing that, to me would be interesting for us to think about as a field, is it possible to, move the patient earlier into the development process to understand the potential benefit? Again, along these lines of perseverance, are there certain intractable syndromes, for which the treatment alternatives are maybe high dose opioids, or maybe some neuroablative procedure, or maybe some invasive procedure, where the risks are very high, just as we think about, in other disease areas. And is there an opportunity to think about how, we could get a type of human validation in patients, not in normal healthy volunteers, to give us an earlier sense of confidence that we should move forward with a mechanism? I don't know, Anna, do you have thoughts on that?
I mean, the NCATS, for example, does have an interesting FOA for people working on gene editing. And when the grant concludes, the company has to provide, or the academic, guidance for others. So I also think it's some of these grant mechanisms at the conclusion, potentially we can work together on providing guidance for others, or what we've learned, right? So we're not, we're using money wisely as well, right? Investment in terms of the government investment wisely as well, as to how can we help others that are going through the same challenges as us without them having to go through the same wheel. Obviously, we're all different. We're all doing a little slightly different, but the more we can provide guidance or learnings along the way, I think it'll help others in the process as well.
Great.
But yeah, and going back to your point on getting, because we're doing gene therapy, we can do a phase 1/2 together. So we have the advantage of being able to not just look up safety, but also early signs of efficacy with a lot less capital. But with small molecule approaches, it's different and it's more challenging. And so, like you mentioned, how can we bring that earlier into the process? And maybe it's going to take a group of researchers to convince potentially the FDA, right? To make more advocacy there.
Yeah, I think that's been transformative for the field of oncology, both in terms of the success rates, but as well as the investment context and the willingness to accept risk. And I think that, again, I'm not aware of, that as a practice in the analgesic development space. And it would seem to me that that's a potential opportunity, especially for, therapies like gene therapy or others, where it may be sort of more of a one and done, or one for 5 years. Where again, the hurdles to get into a phase 2A is much more, prolonged. and so, how do we reduce the, the friction there?
John, can I try to address a question that was in the chat?
Please do. Yes, of course.
Yeah, all right. So I know that many of us are at medical schools. I happen to not be at a medical school. I'm at a undergraduate institution, and the question is about creating opportunities for PhD students to get more experience in entrepreneurship and biotech. So I think for those of us at institutions like mine, this is a really great thing for us to get involved in. So we often have business and management schools that have courses that are put together for people that might be more in the software or tech world, not so much the biotech world, but they're often very open to gearing some of those classes towards students that are interested in this, and potentially PhD students, but also making sure that postdoctoral fellows can get access to these courses. I think that's very important for us to influence our universities if they're not currently thinking about postdocs being able to get into this kind of coursework, to allow them to do it, because that's also a great group of people that might well be very interested in doing this as a career path. So Ryan, to your question, I think that we can do a lot more, and that there are some places that are already doing a really great job. At UTD, we're actually redoing our program now, so I'm hoping that's going to roll out next year, which will be pretty exciting. So lots of opportunity there.
I would like to bring up a point also. I mean, I know there's a whole panel just focused on biomarkers, but I think that just such an important aspect for us is, can we potentially collaborate? People that are developing therapeutics, working with people that are developing biomarkers. Is there a way of working together? That's one of the biggest questions I always get is, is there a way for you to show that you have target engagement, or to show that the patient has a decrease in pain? Is there a challenge study? Is there, imaging? So all these things I think are important, just to show ways that we can de-risk along the process in the clinic as well.
There's one more comment on the chat that's related to the entrepreneurship question, and brings up the NIH entrepreneurship bootcamp. So that is another excellent opportunity, and yet again evidence that NIH is trying to be as supportive in this area as they can. And if I can just go back to one point that both Anna and Daniela made, and I think Daniela, your idea on this was really fantastic. The toxicology and regulatory stuff is such a huge bottleneck, and it's so hard to find people that can help you in those areas. I think the idea of doing this, like something like that with the PSPP, is really an amazing idea, and it would open up so many opportunities for people to move their programs along more quickly.
That would be wonderful.
Well, I think we're at the end of this session, so just want to thank Anna for taking the time to prepare this, as well as Daniela. And I think we're going to come to the last component of the day, which is an open discussion of areas of interest. I know we have quite a few people online as well, so look forward to hearing from all of you. So Ted, do you want to open up and take any questions here?
Sure, but I think we first have a break.
Oh,
So
perfect.
yeah, we got 10-minute break.
10-minute reprieve.
Yeah,
Fantastic.
yeah, and we do have a discussion to come. So yeah, maybe we have some questions I think weren't addressed in the chat that we can come back to. But if y'all have questions also during the break, you could go ahead and post some of them, so we can start with that. And I think we're gonna open up the mic for people also that are in the audience, right, to chime in. So also
for the attendees, this is really your chance. We have a good chunk of time here. There's been a lot discussed, and now is a real opportunity for folks to kind of pitch in on the conversation. So take your break, but please come back.
All right, great. Thanks, everyone. See you in a little bit. 10 minutes.
Okay, everybody. It is 12:10 Central Time. I think it's time to come back. See, John is back. We're back. Great. So we're going to stick with folks typing their questions or comments into the chat, and then we'll address them and or read them to avoid any kind of technical issues we might have with moving people from being attendees into the panel. So if you could please either type your questions into the, or comments again, into the chat or into the Q&A. The Q&A makes it a little bit easier for us because we can keep track of what we've answered or not. But maybe we can start with a question that is in the chat, that is the one that was typed in while we were taking our break. So this is from Veronica Hong, and the question is if folks have thoughts on involving philanthropic organizations to fund entrepreneurial efforts in pain and therapeutics discovery, and should this approach be proposed or coordinated with NIH? So I'd love to hear if anybody has thoughts on that one.
So there are some patient advocacy groups, some of which have small amount of funding. Some fund, for example, research in migraine or trigeminal neuralgia, typically, for example, the Face Pain Association. However, they're small amounts of money, and so I do think it's difficult to find organizations in pain that have a lot of funding. That's been my experience. We personally did receive actually some investment from a philanthropy group that is focused on difficult to fund ventures, and some of which obviously is chronic pain. They also work on other environmental issues and so on. But again, these are smaller funds, but it's definitely good it's a good suggestion for sure. I mean, obviously.
Anybody else have thoughts on that one? So I'm going back through the the questions that we didn't answer previously, and I see one from Daniela Minichella, and this one was originally addressed to Daniela Salvamini. And the question is, how can the HEAL Initiative assist basic scientists in designing preclinical studies with comprehensive outcomes to analyze various evoked and spontaneous pain modalities? Establishing a comprehensive battery of behavioral studies can sometimes be challenging for a single laboratory. So Daniela, you kind of addressed that one a little bit, but maybe you could go on in a little bit more detail about that one.
Well, I think, the PSPP is a great platform that provides a comprehensive battery of tests that not everyone has in their own lab. So I think it's a great platform, and I don't really have much else to add to that. And I would encourage anybody that has interesting molecules that are semi-validated to send them over to that initiative.
Okay, we don't have any more questions, but maybe I'll just pose a general one to folks here, and people that are listening also can feel free to chime in with comments on this one. So part of what we're doing here is strategic planning for the next phase of the HEAL Initiative, and we talked about some successes and some ideas, but I thought maybe it would be nice to hear from the panel, having heard the talks and other people's viewpoint, if you have some ideas for things that you think should be prioritized, in particular with respect to therapeutic development. And maybe since I'm asking the question, I'll also start. So this is actually something John and I have spent a fair amount of time talking about. So I think we heard about gene therapy. we heard about devices. We talked about small molecule drug discovery, but there are also other modalities, and I think we've all seen that there are big opportunities for therapeutic development around mRNA and other types of oligonucleotide-based therapeutics. And my personal view is that there's also a really big opportunity for targeted delivery of these type of therapeutics to the right kind of cells. The peripheral nervous system, of course, is what I'm usually thinking about from that, but there could be other ways to view this. So I think one big opportunity is kind of broadening out the modalities, and this one is an amazing one because from the mRNA therapeutic type perspective, we've obviously seen the amazing success that this has had getting us out of the COVID-19 pandemic. But the technology there, was developed long before the HEAL initiative started, but it wasn't really therapeutically implemented until after the HEAL initiative had already begun. And it might be a fair assumption that all of us on this call have received an injection with that therapy at some point in our life now, which is kind of amazing that we've gone, from it not existing when we started this effort a couple of years ago to it being a totally transformative type of therapeutic modality. So I think that personally that's a huge opportunity, and that we're just starting to learn about how we can take advantage of that particular therapeutic modality to get targeted expression of genes and cells or tissues of interest.
Yeah, I just want to underscore that. I think that's a great point. To me, the nucleotide-based analgesics clearly is a direction that we're ultimately going to go, just like so many other therapeutic areas have already advanced to in cardiovascular disease. I was thinking of that as I was listening to David's talk at the beginning and thinking about statins, but thinking about when you look at the future landscape of cardiovascular disease, clearly it's you see the coming of nucleotide-based therapies there, and I think that there's no reason and that it's not going to come to pain as well. And the sooner the better. Obviously, a lot of safety margin. We heard from Christy and Will about the importance of reducing the unwanted side effects, cognitive, weight gain, sleep disrupt. The whole panoply of problems that come with centrally acting analgesics. And so I think that to the extent that a nucleotide-based analgesic delivered either subcutaneously or through even the intrathecal compartment is something which could selectively knock down a protein expression along the axon or distally or in the DRG, seems to me to hold enormous promise to kind of meeting the goals that Christy and Will set out for us, which was, to minimize the off-target or the central effects of therapies that are frankly unwanted and make the trade-off decisions for patients seeking relief, so dire. Right? You shouldn't have to choose between, not being able to focus in a courtroom or gaining weight or, not being able to do some other vital task in your life, like care for your children, because you want to have pain relief. So to me, the potential here is to, work hard on a class of therapies, which may be a runway to avoiding that trade-off.
I'd like to bring up the idea, being in the biospace and biotech space, a lot of times AI comes up and computational models, advanced computational models come up. And how can we implement those to help us, right, when we're trying to develop non-addictive therapeutics for pain? Some people are using AI for patient stratification, for, in general, to actually obviously design drugs and medications. So is there a way for us to think about using this to streamline this process, right? Or funding mechanisms for people that are experts in this area to bring their expertise into pain could be interesting as well.
Any other thoughts?
I also wanted to quickly comment about the side effects and different behavioral tests. I think that is also a similar situation as Anna just mentioned. So sometimes specific labs won't have that expertise, and that could be a weakness in a grant review. But if we have this reinforcement from NIH that we could partner with people who have that expertise, but they are not from the pain field, I think that would be really helpful for us, because sometimes adding someone not from the pain field could be not well seen as well. So if NIH really encourage us to look for more collaborations outside of the pain field, but that could increase our ability to evaluate those different behaviors and different modalities that are not our expertise, I think would be really helpful.
Critical point. I personally think another theme for us to explore, to think about, and I think that would be wonderful to have this sort of writ large in the mandate for the next version of the HEAL initiative, is just to keep in mind that, oftentimes the mechanism of nerve injury is importantly decoupled from the chronic pain syndrome. And the facts are, I think is that, the more we sort of articulate and view pain as the underlying disorder, which may be importantly disconnected from the genesis of the problem. And I think that's, obviously this is slightly different from the scientific question, which we heard earlier, because I understand that it's essential for hypothesis generation and studying mechanisms. But at the same time, I think that it's in clinical practice, we realize that, pain is the disease in many patients. The nerve injury is over, or the therapy that we're giving is going to only modulate pain intensity. It's not going to prevent nerve injury. It's not going to reverse nerve injury. It's only going to modulate pain intensity. So I think that the more we bring that understanding and insight to the kind of therapies we develop, the broader populations who could potentially benefit. Because if we're really talking about a therapy which modulate hyperalgesia in the peripheral nervous system, again, that cuts across, the osteoarthritic pain that Anne-Marie talked about, or the neuropathic pain that Daniela was modeling. And I think that the sooner that we could get to that, in a world where, access to these therapies is only going to grow more and more constrained by your inclusion criteria of your clinical trial. So I just think ways to reconceptualize, the underlying condition as a pain condition, I think is just an important foundational theme, which it sort of hopefully can spread throughout many of the HEALS programs. I don't know how others feel about that, but I think to me that's something which, frankly, could open the door to a lot of possibility.
So we have a question in the Q&A that is also kind of related to that. So maybe I'll go ahead and read this question, and then folks can comment on that and also on John's point. So this is from Richard Slivicki. There's lots of talk about the importance of human tissues for target engagement and general learnings. Given the large gap between preclinical and clinical efficacies, are our basic research efforts better placed on optimizing distribution and assay development in human tissues? Is it still advantageous to put our efforts into generalizing better measures of animal pain endpoints such as grimace, spontaneous behaviors, machine learning, etc.? Or are established pain behavioral assays such as the Von Frey or the Hargreaves test better used as checkpoints to then move on to human tissue validation, expression, electrophysiology, etc.? And he's wondering from a therapeutic development and regulatory standpoint. And I think that's a very interesting question. So we have thoughts on that one.
Sorry, are you, I don't know, is it in general or
For anybody, yeah.
Sorry, I lost my train of thought.
It was a long question. Can you summarize it?
It was.
Can you
Yes, sure.
summarize
So
it?
I think it boils down to what is like the best path forward? What do people think the best path forward is for identifying a target and then moving towards therapeutic development? And I guess embedded in there is how can we get better human validation for targets?
I think pursuing things with human tissue in terms of cell lines, et cetera, I mean, that's something that can easily be done in parallel while you're looking at mice. Those things are not mutually exclusive, and they don't have to happen in series. They can happen in parallel. Once you identify a potential target, you can look at its expression in humans. You can look at its interaction with the drug at that particular protein, ion channel, or receptor. So yeah, I don't think it's one or the other choice. I think where it comes into is, everything has its limitations. Yeah, mice are not humans. But since I guess I'm a believer in evolution, I believe that many of those things will be conserved. But, once you have a drug, there are going to be differences in metabolism in humans too. So there's no way to say that if something works really well in a mouse, it's going to work in a human. But on the other hand, studying neural circuitry and and higher function things is simply not possible in humans at this point. And so I think you have to, use the best of each system to get the information that you can. And, one doesn't necessarily have to make a choice. But certainly when you get to a certain point in the mouse and you think that a drug has, or a pathway has certain potential based on the assays that we know, and I think there's always room for better assays in terms of behavior. There are people on this Zoom who are much more experienced in that than I am, but there's always room for, a mouse is not a declarative animal, so there's a limit to what you can judge from that, but there's always room to get more sophisticated readouts. But, once you get a readout, then, that doesn't preclude, going into people. It just depends upon when a drug company decides it's time to do clinical trials. But, even if something looks good in a mouse, you don't know how it's going to work in humans. But I think the animal models are sort of essential to trying to, do that mechanism-based part of doing a first triage to know whether you're sort of on the right path. Otherwise, there is no way to test this in a human other than clinical trials, right? You can do that, but the cell work can be done at the same time in a human system or in a rodent system. There's no reason to have to make a choice at that point.
I was actually really surprised that Vertex did not do any animal model work, right? They went straight into human cell lines, primary tissue, and NHP. But I agree that, in my opinion, it is important. And the more we show across different types of pain that our molecular drug works in H mice, in female and male mice, for me, it just gives more confidence. But also, I would say to add that diversity when we're using human tissue as well, different age groups. A lot of times they'll give tissue for younger patients because, again, it does when they're in culture, right? Primary DRG from younger patients do better. But we've strived to ensure that we're covering, 80-year-old patient cells, male, female, everyone, because again, there's just a lot of diversity here. For me, it's about de-risking in as much as possible, so that when we're getting to the clinic and we think we're going to help the patients that we think we're going to help.
Annie, you know more about this than I do, but I suspect that Vertex went straight to humans because there had already been a lot of validation of that target from human genetics as well as from some pretty intensive studies in mice. So that would be my guess, but you probably know more about that than I do.
No, that's a really good point as well. Obviously, it's a validated target. I don't know. I still was expecting maybe to look into PK/PD, like, okay, what dose works and how do you translate that? But still, it's like a good point that it's definitely a validated target.
Yeah, I think part of the issue ultimately with Vertex was that their NAV inhibitors didn't block the rodent channels very well. But I think David is absolutely right. It was already very thoroughly validated by Markman and John Wood and others. So
I would
Anne-Marie.
I would also I think that there's there should be probably ways of concentrated effort on finding out how we can get quicker to human testing. And for example, I say that because we have been pushed so hard in mice to do everything in male and female mice. And I think personally that a lot of that is a waste of time and effort. For example, in osteoarthritis field as a whole, not just the pain, females get a lot more symptomatic OA than males. And all our mouse models are predominantly male because the female mice don't get much OA. Now, that might be interesting thing to study in itself. But to then just say, oh, let's do everything in male and female mice, I think is actually not useful. And so I would make a push to it came up in the chat here the whole time. How can everybody from Anna, from the company to everybody, how can we access human tissues? How can we? So I think that's a big opportunity also for us to come up with ways to make that more effective or efficient.
Yeah, something that similar potentially to the whole small molecule screen, the iPSC work that we collaborate. So these different efforts potentially having instead of iPSC core, more primary tissue core, where we can collaborate with NIH potentially. They might have better access to these tissues, or if not, ways to develop a network to help grantees have better access to these tissues, I think would be really helpful. We do get access, but it's very sporadic, and sometimes we don't even get a DRG, we just get nerves. So yeah, it's also issues with quality control sometimes as well.
Mhm. Yeah, I think also going back to the animal model discussion, I think that's one of the biggest opportunities for machine learning actually, is to use those kind of approaches to be able to look at pain suppressed or pain modified behaviors. And of course, there's already a lot of really great work that's happening in this area, but I'm sure as the AI explosion continues, that this is just going to get even better. So I think one of the mistakes that we make is trying to interpret, assign our own interpretations to animal behavior, and doing it in a way that is completely unbiased, like we do with almost all sequencing experiments nowadays, is a really cool way to think about doing that, and that it's likely to lead to major improvements in our ability to understand what is actually happening in the animals and some of the models that we currently use, and also in designing new models. Any other thoughts on Richard's question, which nobody may remember anymore since that was some time ago? But we have a couple more questions in the chat, which maybe I'll go to. So the next one is a comment and a question. Actually, it's mostly just a comment, but from Daniela Minicello. Thanks for organizing the Heal Pain strategic planning session. I want to emphasize what was mentioned at the beginning of the webinar regarding the importance of targeting the periphery to aid the development of topical therapeutics. This approach aims to minimize the side effects that patients often struggle to tolerate, as described by Christy and Will, and also based on what they described in their personal experiences. So I think that's a great point, and it's another modality that we don't talk about very often, but topical therapeutics. I see Cheryl Stuckey's on the call. Daniela, I know, has done a lot of great work on this, understanding how cells in the skin communicate with nociceptor endings. And Marie talked about the same thing in the joint. David talked about it, and the viscera, extremely important. And the skin offers us a great therapeutic opportunity. Anybody else care to comment on that one?
I was also going to point out, I totally agree with that comment. But something that more groups are doing now is to look at the central mechanisms of peripheral interventions. So I think we should be more open to this idea of it's not really just the periphery or just the central, and we are kind of two fields that are fighting each other. So actually, we can see how both systems would interact, and maybe through the periphery, we could develop better therapeutic targets that would also have an effect centrally, right? Indirectly, but also have an effect. So just wanted to bring that to everyone's attention.
I think that's a great point. Also, while we're riffing on routes of administration, I also want to mention, I think, the importance, and I'm sure Prasad could speak to this perhaps more eloquently than I, but the importance of intrathecal routes of administration and epidural routes of administration, and I think this is I think often perceived as sort of a major barrier, sort of when folks outside the field think about this, but the reality is that there are so many patients across the country, 15 or 18 million every year, who choose to have quarterly or regular epidural or other intraspinal approaches for treatment of their pain, and these allow us to access the central compartment, oftentimes in a way that has a gradient, so you're not seeing the central effects or having most chronic pain problems, or the majority are focal and segmental. And so I just think that as we're thinking about development and making investments, it's really important to explore these routes of administration because we do have an infrastructure of clinicians, set up to deliver these therapies. And, I think, for many patients, these are really, life-altering benefits. And so I think that the extent that we can take more sort of, really novel modalities and put them into these routes of administration, I think that could be a major advance because, we're sort of using medications mostly from the 1950s or the 19th century through most of these routes of administration or earlier. And I think that it's been sort of disappointing that, in the last 30 years, we have not seen a pipeline of new assets that, could go into the central nervous system, and I think provide relief there. And I just think it's a real opportunity and one that I, have encouraged, folks in industry to look at, but also, colleagues elsewhere, because I think that these are things where, it could make a difference for patients sooner. And even, these procedures, while they are invasive, they're often incredibly well tolerated and they're very easy to do for clinicians who do them routinely. So they're not as daunting as they might seem to someone, frankly, who's never been exposed to them. I don't know, Prasad, if you want to comment on that.
Yeah, thanks, John. Yeah, you bring up a really important point. And specifically, I think intrathecal therapy, being able to infuse drugs directly into the spinal fluid, we have the technology to do it, but you're right, it's vastly underused, right? So the drugs we use are old. Most of the drugs we use clinically are not the FDA approved ones, because there's only 3 really, 4 drugs that are FDA approved, but 3 for pain. And it's basically morphine, baclofen, ziconotide, and how do I put this? If we could develop an mRNA target, or if you can develop any drug that needs better penetration, and we already have a route to administer it. So you're totally right. Using something like that, using that existing route to deliver medication would be phenomenal. One of the biggest challenges for epidurals, nerve injections, joint injections, is they work, but they're transient, right? And so durability is a huge issue. One of my biggest wish lists as a clinician is if I could inject something into a joint that would last 6 months or a year, my patients will be way happier. There's less financial incentive for physicians, but who cares, right? Like that is a huge incentive for insurance companies and I mean for patients. So I was very excited about some of the initial work on. I guess there were some funded grants for DRG and like an injectrode, or using devices that are injectable. But I would love to see right now, like, how can we increase the durability of even existing compounds that exist out there? Right now we use steroids and anesthetic. There's this drug, Exparel or liposomal bupivacaine. It lasts a few days as opposed to a few hours. It's still not great, right? We need something that lasts safely months or up to a year. So I think that would be an excellent focus for therapeutic delivery. I guess it's more of a kind of maybe a chemistry solution, but a material solution, but increasing durability of existing compounds would be awesome.
Yeah, and I would just add to that list ultrasound. This is, so we're living through the golden age of ultrasound, and it's underwritten an absolute revolution in perioperative analgesia, and I think it has the potential to do it. We have the right therapeutics to deliver. The ability to target now, the neurovascular bundle is extraordinary, given the imaging resolution. And I think this is an area which is, sort of been underdeveloped in terms of the chronic pain field. It's certainly been leveraged in acute pain using older analgesics, but I think novel analgesics delivered through ultrasound guidance to peripheral structures like a ganglia or even a peripheral nerve, I think holds real potential.
So we had a little bit of a biomarker discussion, or that there were some questions about related to biomarkers, and I just wanted to point out, because Rob also brought it up in the chat, that we do have another group that's dedicated to biomarkers, but I think they already did their session, so maybe we could just bring that up really quickly. I know it's come up a couple times. I think in the context of what we're doing, what we're talking about today, one of the really important things is having a measure of target engagement in the clinic. So I'm sure people are at least somewhat aware of some of the NaV1.7 inhibitor trials where I guess that there was this initial thought that maybe the inhibitors lacked efficacy. Raj Khanna also brought this up in the chat. But I think we know now that that probably was not actually the case, that the initial some of the initial NaV1.7 inhibitors that were developed had poor pharmacokinetics. There's a really nice Science Translational Medicine paper on this from the Merck group, where they describe how one of the initial compounds, it's a PF compound, I think that means it's a Pfizer compound, had poor pharmacokinetics and may not have really reached the target. And that was one of the reasons that it likely didn't have efficacy. So that kind of highlights a major issue in clinical development, is that, you know, we're thinking about targets and efficacy in animals, and it's pharmacokinetics don't always equate from one animal to another. Metabolism can be totally different in one species versus another. It's not super common, but it certainly happens. And one thing that I think everybody would like to know when they advance a molecule into the clinic is, how do you know that you actually engage the target? Now, for some things, you know, you can use PET imaging. That's a pretty complicated way to do it. So sometimes you can have a biochemical measure. For sodium channels, I think we've all seen the amazing work that Jordi Serra has been doing related to microneurography as a good way to understand whether you've engaged the target, but it can be very challenging. And I think for every new target that people are starting to move forward, that's a really critical piece to think about. We certainly had these discussions for our UG3, UH3 grants, because there's so much emphasis on it as you move into the clinical development stage. So I think that's one very important aspect of biomarkers that is a little bit different maybe than the way that biomarkers are typically discussed in the pain area, where we're often trying to have a better set of biomarkers for understanding mechanisms in patients and stratifying for clinical trials, or maybe even having a biomarker for pain in general, which I know is a topic that has come up a lot.
Yeah, I think that Ted, to your point, I mean, looking at you know, skin PD, you know, skin knockdown of certain proteins on punch biopsy obviously is a way to look at, you know, protein expression. You know, one possible avenue there, and to sort of refine those types of endpoints, you know, I think is really a key roadblock to really to think about how to tackle. You know, because I think that, again, that's, as going back to our point about the mRNA or nucleotide based, you know, therapeutics that affect protein expression. I think that, you know, refining those relationships there in terms of the time course and where you expect to see the knockdown, and how that knockdown correlates to trafficking of a protein across an axon, and how that relates to pain intensity. I think working out some of, you know, those details and public sector investment in those types of questions, I think could really, you know, open up opportunities that, you know, to give folks more conviction that they're in their, you know, they're engaging the target and they're seeing a readout, you know, that's meaningful or potentially meaningful in patients.
We have another comment in the chat that I'll read and folks can address. Says, most rodent models and outcome measures in pain are mainly focused on the biology of nociceptive processing and resulting behaviors, but not necessarily chronic pain. The emotional, subjective, individual health, and comorbid or co-occurring health condition components are missing in these models and are difficult to assess. As mentioned earlier, emphasis on reverse translational approach to model human pain conditions and associated diseases and comorbid conditions to understanding the biology of ongoing human pain conditions are the needs for the field, which have been ignored or sidelined for decades. So would anybody like to address that one?
Not to hog the mic, but I'd love to just pick up on the point that Christy started with today, because I thought what was particularly poignant about her discussion was, you know, the way she described not only her headache pain, not only her musculoskeletal neck pain, but her neuropathic arm pain. And the reality of clinical practice, and I'm sure others can speak to this, is that when you see patients with chronic pain, it's often, you know, all of these systems which are you know, interrelated in the person, which are affected and variably on a single day. And, you know, that anecdote you shared about the, you know, the nurse or the clinician, you know, entering the room and asking you to give a number, to assign a number to the complex panoply of different systems and parts of your body that are being activated on any given day. And to reduce that to a number is just, to me, it's not an affront, but it's obviously an impossible thing to do. And yet you're forced to do it to sort of continue with the visit. And I think that, obviously that's a barrier in practice, but I think it's also a barrier in analgesic development. And I think that, this is not the endpoint discussion meeting, but I just think that, getting to better endpoints is going to be the key to getting to better therapies that work for the holistic experience that patients go through.
Can I ask the question in response to that question, I guess, to the rest of the panelists? I often will see, well, I've had interest in proposing grants or studies where we do human and animal work together in one grant, but I often will see comments, from reviewers that, well, one, it's overly ambitious, that there isn't like a clear relationship, or in fact, that there isn't qualified personnel, to do both of these studies in a single grant. It's really hard, to do both of these, but is there value in such a dual, like a study that uses, I guess using cell lines is easier, but using, in vivo human studies with in vivo animal studies, kind of in parallel? And if so, what might be the proper way to encourage that or support that?
That's a great point, Prasad. I would imagine many of us on the panel and many listening have had the same experience. And again, I think that's an area where the HEAL initiative can play a big role. So, I mean, recommending that there would be calls that would be specific for this kind of thing to, have studies that would be done by experts in animal behavior and also people that maybe do brain imaging or other things working together could be a great way to better identify mechanisms or therapeutics.
Yeah, our goal for this process is to identify the gaps and funding, opportunities to do combinations of human and animal studies seem to be missing. Yeah, that used to be possible, and then there was a specific prohibition against that by NIH, or at least certain of the funding opportunities for a long time. But just to mention that's a gap. There is a perceived challenge, as Prasad indicated, that it's to get not just the right people on the project, but the right reviewers to be able to do that was a challenge. But I think that's not something that should prevent that, because that type of work is so important.
Yeah, so DP Mohapatra just posted something on HEAL RM1 that maybe we should all read more carefully. All right, we have another question in the chat. This one from Lauren Kelly. Any development on meds to treat pain and co-occurrent opioid use disorder? Buprenorphine functions in this way for me as a clinician. Any development on microinduction treatments to make it easier for patients to switch onto buprenorphine from full agonists, long-acting injectables of buprenorphine for pain indications? I'm not qualified to answer that one. John, you're Prasad, or some of our other MDs on the panel have a thought about that one?
Well, I'm not aware of current developments in this area, specifically for helping patients make, this transition. I do think it has been one of the major advances in pain management in the last 15 years is having the availability of buprenorphine to help patients, especially, for example, a patient who was started on an opioid because they had intractable hip pain before a joint replacement. They developed a tolerance to that medication over the course of months. They had that surgery. Their pain intensity is reduced, but obviously they're now dependent on their opioid analgesic. And I think that, they're trying to step that patient down off a high dose of morphine sulfate, after that surgery, 10 years ago was incredibly difficult and tedious and painstaking part of practice and really a hardship for patients. I think buprenorphine has made that transition, so much easier, because of its unique pharmacology. But I do not know of a sort of a single magic bullet yet to smooth out that transition. But I do think that it is a lesson to me in the importance of refinements of opioid pharmacology, because I do think, buprenorphine obviously is far safer for many patients and does allow them to reduce their opioid use in a graduated way without a lot of the side effects. And so I think that, this is about developing non-opioid analgesics, but I do think that, one key element of this will be, helping patients, finding therapies which can help patients, as this questioner asks, reduce their opioids in a way that's frankly less challenging to do. Because I do think there are many situations in clinical practice still today, and Prasad, you can speak to this, where, an opioid is the treatment of choice, right? Everyone who has major surgery in this country has an opioid. They have fentanyl, basically. And so the question becomes, how do we help patients, especially vulnerable patients, transition off of that in their perioperative period? And so I think this is a key question, and an area where we really need to have adjuvants or other therapies developed, which will help patients make that transition, because 30 million surgeries a year, it's a certainty that many patients will be exposed to opioids, from those procedures, and they will need a way to come off these medications safely.
Yeah, thanks, John. I would add, I totally agree. In addition to, using opioids more judiciously, or changing our implementation, our practice around how we use opioids, partial agonist opioids like buprenorphine have been, amazing for me as well clinically. I use them all the time for pain, or I use buprenorphine in its various forms for pain. The patch is great. It's a once weekly patch that you replace. It's become much more accessible as well. The transition is difficult. I think, let's just say, I think things are going to change in the next year or two now that the X waiver has been waived. You don't have to have special training to use buprenorphine. And so, I mean, that's definitely a step forward. But, I remember years ago, there was, for opioid use disorder, there was a particular form of buprenorphine developed in the form of like slow release rods. And I think you basically had to, they're subcutaneously implanted. I don't know if it required an incision or not. That never really took off for pain. I mean, it would be an amazing, therapy to explore. And I don't know why, but I will say everybody, there's a bit of there's no clear, in my mind, guidelines for the use of buprenorphines or for pain clinically or perioperatively. When patients come in already on buprenorphine and they need surgery, often they'll get full dose opioids and tons of them on top of it, and it's kind of common practice. It would be great to even study that, frankly, and have clear guidelines of what to do, because different physicians also have different practices around how they manage buprenorphine for pain when someone has some kind of injury or surgery on top of it.
Well, we're down to our about last 5 minutes, and we've mostly answered the questions, although a couple more are popping up. But I thought maybe while I look at those, I'll give folks from the panel the opportunity to chime in with some closing thoughts. So if y'all have closing thoughts based on everything you've heard today, maybe you could quickly share those. And I won't call on people, so you can chime in if you'd like to.
Well, I think as usual, there's always some limit to resources. And the elephant in the room here maybe is that we don't know what's going to happen in the near future with regard to that. So we're going to have to be pretty strategic about where we think the money's going to be spent best, and to what degree it should be focused on understanding mechanism, to what degree should we be asking for NIH money to be spent to do translational things that perhaps should be more in the purview of the private sector. And those are all going to be interesting discussions that will become, I think, more and more acute in the next couple of years. And so if we want to advocate for, doing sort of preclinical work that we think maybe companies should be doing to decide whether a molecule should move forward, or if we think that's interesting or important, these are all interesting questions, or whether we still think that we need to make bigger inroads on understanding mechanisms so we can identify targets, or methodologies for device design, whatever. I think, those are questions that are going to become more and more, the strategic application of these funds will become more and more pressing in the next few years. So I think we just have to remember that.
Mm-hmm. That's a good point. Anybody else have thoughts they'd like to share?
Well, one topic we didn't cover, in as much depth, but I think it's just really worth talking about because it's sort of the bookend to the osteoarthritis and musculoskeletal burden is really the low back pain burden. Obviously, that's to me, really at some levels, one of the key, sort of clinical presentations where the opioid crisis really began. And again, I think it is important to recognize that. And I think we haven't made a lot of progress in thinking about chronic low back pain syndromes as discrete clinical syndromes. We've sort of just kind of defaulted to very nonspecific low back pain. And I think to the extent that we make investments into trying to understand this, I think the HEAL initiative has been very important in doing this in terms of the backpack initiative. And I think I just want to encourage the continued work to try and parse the different clinical presentations of neurogenic claudication and lumbar stenosis, which happens in older Americans, radicular pain, which happens oftentimes in older and middle-aged Americans, and then chronic musculoskeletal axial predominant low back pain, which occurs across the lifespan, and axial predominant low back pain, which is really a centralized pain syndrome. And I think, the backpack work to try and parse and understand that, I think is foundational to developing non-opioid analgesics. And I would just encourage that work continue both at the basic level as well as the translational level, because I think that until and unless that unmet need in chronic low back pain is met, the specter of another opioid crisis will loom large. And so I really think that that should be one area, to David's point about where we should think about strategic allocation of funds to understand a problem which no one part of the sort of the entire human endeavor can fund and take care of.
That's a great point, John. And maybe I'll just chime in on another HEAL initiative, the Precision Pain Network. There's been a lot of talk today about human tissues and better understanding the biology of human nociceptors and human pain circuits, and that really is the point of that. And a huge emphasis of that is to make all that data available so that these experiments don't have to be repeated by others, and they can use that information to make decisions about prioritizing targets and hopefully also building a better understanding of why certain types of chronic pain disorders, in particular neuropathies, develop in people. So I also would advocate for that continuing. And
I think with that,
yeah.
I'd love to just, I know we're coming to the end here. First of all, I just want to make sure we take the time to thank the panelists who've been meeting regularly on this committee for your dedication to this. I think it's just very impressive. And obviously, also to the enormous number of staff members from Dr. Koroshetz to Jackie Ward and Rachel and so many others who really put this day together. This has taken an enormous amount of work and forethought, and just want to express my gratitude. To me, it's really heartening to know that, our public institutions are focused on important problems like this, which touch every American and every citizen. And I just want to say it's a privilege to be a part of it. And I can't thank enough the officials and others at the NIH who are supporting this work, because I think it's critically important to improve the lives of everyone.
I think we should end on that note. That's perfectly said, John. So thanks everybody for being here today. And thank you for all the input. We will continue our work on this. And if you have more for us, please let us know.
Thank you so much.
Thank you so
Thanks, everybody.
very much. Bye. Thank you.
Bye-bye.